All right, here, let me start the recording. As we dive into the respiratory system, I just want to remind you guys that you're in A and P, right? Where anatomy determines function. And so I just want, and you know, for the heart's so small. It's basically just relative to other organs of the body. It's just this heart with a couple of tissues. So the overall anatomy of the heart isn't too complicated with the different chambers and stuff like that. Here we're going into a respiratory system. And everybody take a nice deep breath. You deserve it. Take a nice deep breath in and a nice deep breath out. Where did you guys start breathing from? I'm just asking this in chat. Where did you breathe from? Your nose or your mouth? Good. You guys have done some meditation practice. Yeah, when you take those nice deep breaths in, try to use your nose. So there's a couple reasons why. You're going to learn more about it. But breathing through your mouth, oddly enough, right? The mouth isn't actually part of the respiratory system. So the mouth is part of your digestive system. And those two systems will be linked at the back of the throat, but your mouth doesn't have the exact same function that your nose does. Obviously, structure determines function. Your nose and your mouth don't look anything alike. But yeah, take that nice deep breath in. And again, focus on breathing through your nose and using your diaphragm. And then everybody breathe out. So where's the first place that air passes through? Don't tell me nose. I want you guys to be more specific. Where's the first place that air passes through? Come on, guys, flow charts. You know how this goes in my class. Where's the first place that air passes through? Yeah, the nostril, right? The nose picker. Does that make sense? Nostrils are called nares. Any opening in this respiratory system, we're going to be calling a nare. There's internal nares and external nares and that sort of stuff. And I just want you guys to understand that each one of these structures is important. It's not just I'm throwing this crap at you for poops and giggles, right? The nostrils are lined with something called a nasal vestibule, which has hair. I'm just getting you guys thinking about this already. But what do you guys think all of that hair in your nose is good for? Don't tell me nothing. Don't tell me that it's gross. Most of us trim our nose hairs, but it serves a function, and it's a really important function, and that's filtration. Exactly. It's filter. It catches all the bad things. I love that, Heather. It's exactly the words that I tend to use. Nice, simple words, right? Catches bad things. Gets rid of dust particles. But everybody do me another favor. Everybody take a nice deep breath in. Congratulations, you just breathed in about 1 million bacteria, right? So we have a filter in our nose to try to catch things like bacteria that's in the world that's out there trying to harm you all the time. So as we go through the rest of this respiratory system and the anatomy of the respiratory system, primarily with the focus of the conduction of air from the nose down into the lungs, I want you to be thinking about what each part of the anatomy is good for. Okay, what physiology is it designed for? Is that clear? Does that make sense? Awesome. Cool. Love it. Love you guys for working your way through that. Now, I also want you to look at the overall respiratory system real quick, and I want you to think about what is the big picture function of the respiratory system. This is a little bit more complicated than you think. So let's go ahead and start chatting about it. Get O2. I love it. Melissa, that's most people's conception. O2 in, CO2 out, great, great. Gas, exchange, nice kind of easier way to figure that out. Good, all good. Isn't there more? Didn't we just discuss something that's kind of cool about the respiratory? Everybody take a nice deep breath in. Where does gas exchange occur? Filter, but we're going to come back to that. Where does gas exchange occur? Gas exchange from the respiratory system, I guess I should say then, because you guys are focused on the blood. You're not wrong. It's occurring at the blood, but it's occurring inside the lungs. And Heather, perfect. It's occurring in the alveoli. So it's going to be an exchange between the air in the alveoli and the air in your blood. Yes, ravioli. I love it. Hey, did that make sense to everybody? That gas exchange is occurring in your lungs. So does air magically get down into your lungs? Is it magic? Or do we have an anatomical conduction system that allows air to travel through it? True story? Does that make sense? All right, so now next step. I know we always do this every beginning of class. I just want you guys, I'm not jumping right into the lecture, right? It's just not my style. I want you guys thinking about what the unit is about first. Take a deep breath in. What's the first thing you did? How did you just breathe? What's the very first thing that happened? Oh, love it. Yeah, you inhaled, but how did you inhale? And a couple of people threw out the answer, okay? You inhale by contracting your diaphragm. Crazy, bizarre that that is the initiator of this breathing process. One big muscle and a bunch of other smaller muscle groups, but the big muscle that's involved in your breathing process is called your diaphragm. Now, focus on your diaphragm, right? Remember, this is this huge muscle that separates your thoracic cavity from your abdominal cavity. It's this really huge muscle. Focus on it and focus on your thorax, your thoracic cavity. As you breathe in, your diaphragm's contracting, and what's happening to your thoracic cavity? Focus on that. Talk to me. Oh, it's expanding. That's perfect. Did everybody feel that now? Maybe you didn't get it the first time around, but everybody kind of maybe even put your hands right on your thorax, right on your chest, and then take a nice deep breath in. Do you feel that chest expanding outwards? Guess what? It's not air rushing into the lungs that's causing that chest to expand outwards. Okay, it's muscles that's doing that job. And you guys are going to be learning some really basic physics that explain what's happening and how muscles initiate breathing, and that changes the volume inside the thoracic cavity. And because the volume changes inside the thoracic cavity, it's going to change some pressure gradients. And when those pressure gradients change, we're going to start to create vacuums. And those vacuums inside your thoracic cavity are absolutely critical for lung function. If you break that seal, if you break the vacuum that's holding your lungs open, your lungs will collapse, and this is called a pneumothorax. And we're going to go through all of that as we go through this system. Okay, it's really cool. We got to think a little bit more than just breathing. We got to think a little bit more than just breathing. You might see my face, still not very awake. We'll see. Ooh, camera worked. Okay, so functions of the respiratory system now. If we needed to write the very first function of the respiratory system, it's not going to be the first thing that happens. The most important thing's gas exchange, but the first thing that happens is not gas exchange. True story? The first thing that we just explained happens is called conduction. Okay, it is a type of gas exchange, but it's a gas exchange between the environment, the external environment, and the internal environment. And we need to, excuse me, we need to be able to conduct air through a set of tubes. Just going to ask, I guess, does anybody see a link between what we just said and something that we talked about in Unit 2 with cardiovascular system? Is there a link, maybe a key word? Because in Unit 2, we talked a lot about tubes, right? We talked a lot about blood vessels. True story? And as we moved liquid through blood vessels, what did we call the movement of liquid through blood vessels? We called it flow. We called it blood flow, right? And in physics, that's exactly the word that's used for the movement of material through tubes. Here, not blood. Here, it's airflow. Does that make sense? So as we conduct air through tubes, this is flow. Don't we already know a shit ton about flow? What's flow always going to be driven by? What drives the movement of material through a tube? High to low, which tells us what? What formula did you guys learn for blood flow? Delta P. And then it's traveling through tubes, and the tubes apply resistance. And guess what? Airflow is just not different at all. It's the exact same thing. We're going to create a pressure gradient. That's what our muscles are for, and that's what the vacuum inside of your lungs is for. We're going to be talking about the resistance of air as it flows through tubes. What's the number one way that we can change resistance through a tube? Because resistance is proportional to 1 over r to the fourth. So we're going to be changing the radius of our bronchioles. These are the little tubes that travel down into our lungs themselves as they lead towards the alveoli. So you guys don't have to magically come up with a whole bunch of new information. The base for airflow is already in your head. You just have to determine versus just cardiac output creates the pressure gradient. Well, that's not how the lungs work, of course. We're going to have to look at something else that creates that pressure gradient. That's a little bit more complicated. Resistance, easy peasy lemon squeezy. Okay? 8 times the length times viscosity divided by the radius to the fourth power. Can we change the length of our airway? Just to think about this real quick. Normal physiology? No. But has anybody ever gone snorkeling? Do you guys remember that very first time that you went snorkeling and you put that extended tube in your mouth and you're trying to breathe underwater? Does anybody remember that time? You felt like you were suffocating, probably. Like you're having to breathe super hard, and sometimes people panic and just don't want to do it. Well, you just increase the length of your tube, therefore you increase the resistance flowing air through that tube. So you have to breathe harder, and people just don't always like that feeling. So viscosity of the air. Anybody ever walked into a spa right? Where the air is like 100% saturated. It's not just 100% humidity, it's actually saturated, so the air is thick. Yes, no, maybe so. You know what I'm talking about? It's basically Houston or Florida. And the air can feel really hard to breathe because it's so viscous. All right, not to mention mucus and all that sort of stuff. So we'll be talking about each one of those variables more. But once we conduct the air through those tubes, whether we're getting them down into the lungs or whether we're having it leave our lungs, either one, we primarily end up focusing on the, what happens as air is flowing down into the lungs. But now that we have air into our lungs, now we get to have gas exchange. True story? So the next major function of the respiratory system is gas exchange. But this gas exchange is occurring in a very true story I just made up. That's what I do. This gas exchange is occurring in a very specific spot. It's occurring between the alveoli and the capillaries. So we're going to call this alveolar gas exchange. We're going to be learning just a couple more words here in just a second. We'll come back to gas exchange versus airflow. We're going to be using some actual physiology words to describe these things in just a second. Gas exchange occurs. And now, from the alveoli, it goes into the blood. Now, let's remind ourselves about oxygen. Is it polar or non-polar? I always want to remind you guys of this. O2, oxygen double bound to an oxygen. It's like one of the most non-polar things on the face of the planet. Okay, it's tiny, it's extremely permeable, it acts on diffusion. So alveolar gas exchange is extremely likely to just, rush, not even just, there's no thought process whatsoever. Oxygen just moves across our membrane. It's awesome. But it's still driven by diffusion. And when we think of diffusion, we still have to think of the rate that diffusion can occur. Do you remember talking with last week about this? The rate of diffusion in the capillaries, for instance? And the first thing that decides whether or not things diffuse or not are going to be and somebody wrote it up here with high to low, right? What's the first thing that decides whether or not something's going to diffuse? What must you have? Gotta have a gradient. Good. Now, in this case, it will be a chemical gradient. Well, technically, that's not true either. In this case, it'll be a pressure gradient. I don't want to put concentration. I'm sorry for that bracket. Ignore my crazy boomerang. Just gradient works for now. The lungs have an extremely high surface area. We'll be talking about the anatomy of the lungs that gives it an extremely high surface area. Oxygen is extremely membrane permeable. 2 tennis courts, that's right, Tariq. Oxygen is extremely membrane permeable. It's nonpolar and it's very small. You guys learned a quick formula to try to understand membrane permeability a little bit better last week, but really it just has to do with size and polarity, right? The smaller it is, the faster it will diffuse. The more nonpolar it is, the faster it will diffuse. And the last variable is actually to the power. So it's really, it makes the most impact on the rate that something can diffuse. Who remembers what the most powerful, yes, gradients are required for this thing to even exist, but distance it has to travel is going to make a big impact on where diffusion occurs and how fast diffusion can occur, because it's the distance it needs to travel. When we're talking about distance, we're not talking just about the membrane. Good, Melissa, I'm glad I was right in the process of doing that. Remember, it's not just the thickness of the membrane, it's the thickness that oxygen has to be transported across a surface. Now, if there is liquid on the surface of that membrane, and this is nonpolar, and it hates liquid, then that distance is going to increase. Does that make sense, everybody? Tariq got it real quick there. Did that make sense? Cool. But yeah, the anatomy of our lungs, we're going to be talking a lot about epithelial tissue again. Sure, Heather. I could even kind of give you guys a quick drawing of it. If you got a membrane that's real small, okay, like a simple squamous epithelial tissue here, great, awesome. You've already started out pretty darn thin, and oxygen can be transported really quickly across that. But we also have to keep in mind that since the oxygen is extremely non-polar, that it doesn't like water very much. So if we start adding water to the surface of this membrane, then that becomes part of the distance too. Does that make sense? It's not just the membrane, it's everything that gets in the way. Oh, great question. It would be no bueno. It would be very no bueno. It would be not physiologically, or not, yeah, I guess physiologically normal. Water in our lungs is what creates those noises when the doctors are listening to your lungs, if you hear the wheezing noises and stuff. But there's lots of reasons why you can get water in your lungs, and we always have to keep that in mind when we're talking about membrane permeability. Why else do we need to think about it? Because I'm not just talking about the rate of diffusion in the alveoli, I'm talking about the rate of diffusion through the entire conduction pathway. For instance, are we going to have a rapid rate of oxygen diffusion in our nasal cavity? No, because it's not designed for diffusion. The anatomy in our nasal cavity is more for catching and filtering nasty crap out of the air around us. It's also for humidifying and warming up the air as it passes down into our lungs. So when we're thinking about this rate of diffusion and Fick's law, please keep in mind, I'm not just talking about the alveoli. What's so great about the alveoli is they're the ones that promote the rate of diffusion the best. The anatomy of the alveoli, the anatomy of that entire lung system down there, it's promoting the rate of diffusion. But we're going to be talking about other areas of the respiratory system, why that anatomy does not promote diffusion. Does that make sense? Fick's law will help you understand how the anatomy determines the function. Cool. Okay, let's move on. 3. Now we have oxygen in our blood, but it's non-polar. So how are we going to get this stuff down to the tissues that it needs to go to? Heme, yep, sure, absolutely. We need a carrier protein. That's what I was after for those that are kind of sitting there. Does that make sense? That's what I was looking for? I didn't want to go back through the entire, I guess I could have, right? It's an Uber system, right? We got a nonpolar molecule. Can it dissolve in your blood? No. So what does it need to get through your blood? It needs a carrier protein. Uber/Lyft. Nobody uses taxis. Yes, no, maybe so? And this is called transportation. We need to make sure that we can transport the oxygen out to the tissues, and we're going to have to transport the CO2 back to the lungs. Transportation is going to be a lecture all in itself. Okay, transportation is a pretty big deal, and we have to talk about oxygen saturation and CO2 transport. Because what you guys are going to be learning by the end of this unit is that your body actually cares much less about oxygen than it does about CO2, which is a very big, like that idea discombobulates lots of students. Like, what? My whole life I've been thinking O2, 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 but your body has tons of CO2 sensors, and it has very few O2 sensors. But we're going to get that stuff out to the tissues. And finally, don't forget this step, even though you've already learned about this step as well. Once it gets out to the tissues, now we're going to need to exchange oxygen for CO2. True story? So this is going to be a tissue gas exchange. This is the perfect time for me to try to start to define each one of these words with one key physiological term. Normally I would have done this to begin with. I just didn't ever find the time to, or the right place to interject it. But airflow, right here, conducting air through tubes, airflow, this pressure gradient that we end up creating right here. The movement of air through tubes, the physical act of breathing, it has one word that describes it, okay? And this is called ventilation. You guys have ventilation in your house, right? If you have AC, which I think most people in Texas has it, I hope. Be crazy dangerous without it. So as we start moving air through tubes, we call it ventilation. And if we have something that's physically forcing it through those tubes, it's ventilation. Ventilation is broken up into inhalation and exhalation for us as people and animals. And I'm sure you guys have heard these terms before, but maybe didn't know exactly what they were referencing. Inhalation is the act of breathing. Exhalation is the act of breathing. Ventilation is the physical act of breathing. So if you put somebody on a ventilator, here's the way to think about this in terms of medicine. You put somebody on a ventilator. Are you doing the gas exchange for them? Are you doing the transportation for them? Are you doing the gas exchange down on the tissues for them? No, absolutely not. The only thing you're doing is you're forcing air in and out of their lungs. You're forcing air in and out of their lungs. You are ventilating for them. Does that make sense? You are changing this pressure gradient right here. You really can't even direct control of that resistance, although I'm sure there's going to be, some sort of albuterol or something like that in your tubes. So step one, as we conduct air through tubes, airflow is proportional to delta P over resistance. What we're talking about is ventilation. So if on a test I say, tell me everything you know about ventilation, this is what I'm asking you guys to walk through. Does that make sense? Step two, gas exchange, has a word that we use for it. And what system are we talking about here? What is the overall system called? Work with me. I know this is weird. What's the system called? This is the respiratory system. So where does respiration come into play? Well, that's what this is. Respiration is gas exchange. That's what the word respiration means. I find that most students do not know that. So I love adding that in. So when we're talking about respiration, we're talking about gas exchange. So up here in the alveoli, we're talking about alveolar respiration, where there's gas exchange between the alveoli and the capillaries, the pulmonary capillaries. Does that make sense? This is gas exchange. Transportation, we've already learned a little bit about in Unit 2, Unit 1. Ah, Andy, we're on our way there. We were literally about ready. We had seconds to get there. Step 4 is tissue gas exchange, where we're having gas exchange between your blood and your cells. Gas exchange between your blood and your cells. That sure sounds a lot like cellular respiration to me, doesn't it? And that's exactly what Andy just said. This is cellular respiration. You guys might be wondering why this word feels so familiar. Who remembers the formula for cellular respiration? This is the same thing as aerobic oxidation. Take a deep breath in, O2, you eat food, glucose, throw some ADPs, throw some inorganic phosphates, shake and bake, shake and bake. Can't even draw a straight line. Chemical reaction is going to occur primarily inside the mitochondria. What are we going to make? Oh, some good old ATP. What else are we making out of this deal? We're going to make some CO2. We're also going to make some water. And so if we draw this diagram out, what I want you guys to be looking at, and we'll be thinking about the anatomy of our capillaries in a little bit more detail than we did in Unit 2. But what I want you guys to be thinking about is if we have a big, a capillary right here, it's not big by any means. And I have a cell down below, and I want to get cellular respiration to occur. What's happening is that this cell is breathing, basically, right? Cellular respiration. It needs O2. It's going to take that O2 and it's going to convert it into ATP and CO2. Okay, so this right here is occurring inside of this cell. And so what's happening to oxygen consumption inside of the cell? Oxygen consumption. Increase, decrease, no change. Relative to the blood, relative to the interstitial fluid. So if oxygen consumption is increasing, what's happening to the concentration of oxygen inside of the cell every microsecond of every day? As the cell makes ATP, what happens to the concentration of oxygen inside of that cell? It's making ATP, and it needs to use oxygen for that. It's consuming the oxygen. So what's happening to the amount of oxygen inside of the cell as it consumes it? So we have relatively low O2 inside of our cells. Does that make sense? Because it's always using the O2 to make ATP. Yes, no, maybe so? Whereas out here in the bloodstream, we're going to have relative high O2 because your blood doesn't use your oxygen. Red blood cells are unique. They didn't have mitochondria. They don't have a nucleus. They're not using the O2 that they're transporting around. So O2 consumption in the blood is very, very low. Yeah, their delivery service. What did we just have here? We have high O2 in the blood and we have low O2 inside of our cells. Don't we have a gradient? Rate of diffusion is proportional to gradient times the surface area. Capillary beds have extremely large surface area times the membrane permeability. We're talking about oxygen. It's going to be highly membrane permeable, and then all divided by the distance it has to travel. Well, good news. Capillaries are simple squamous epithelial tissue, and hopefully it doesn't have to travel too far between the capillaries and this interstitial fluid, because oxygen doesn't really like to move through fluid. So you want the cells to be as close to the capillaries as possible. Next, what's happening to the CO2 inside of the cells? As it consumes oxygen and glucose and makes ATP, CO2 levels are increasing. And CO2 is also very nonpolar. I mean, it's basically just oxygen with a carbon in between. So it is also highly permeable and will just go from high to low. Simple diffusion. Yes, no, maybe so? And where is carbon dioxide going to be relatively low? It's going to go back out into the blood because it's being transported. And as it's transported away, it makes sure that there's relatively little CO2 out here all the time because it's being transported away. And the fresh blood is coming from this side, right? In this particular diagram, the way I drew it, we got fresh blood coming this way and it's oxygen rich. And then it picks up the CO2 and transports it back to the lungs. So we keep refreshing that system. Does that make sense? That's what maintains our gradients down in those tissues. And this is cellular respiration. That's where that term comes from. It has to do with the cells exchanging oxygen for CO2. I guess we should go ahead and write something down here. Transportation in the blood is all going to be about erythrocytes. And we need to be able to think both about transport of O2 and CO2, just so you guys have it all written down somewhere. Nothing like a good old barter. It just doesn't happen in the United States very often. Can't go and barter too many places. Car lot, I guess. Okay. This is the function of the respiratory system. You can see it's a lot more complex than saying what's the function of the heart, cardiac output, right? There's more going on in the respiratory system. So that's why I wanted to take 40 minutes and I wanted to cover this. But does this all make sense to everybody in terms of breath in? What happens down in the lungs is that gas is exchanged into the blood, then it's transported from the blood to the tissues where gas exchange occurs with the cells. CO2 enters the blood. It's going to be transported back to the alveoli, and then we breathe out, and it's going to flow that air back out into the environment. Then we breathe in, and it's just a cycle. 1, 2, 3, 4, 3, 2, 1. 1, 2, 3, 4, 3, 2, 1. Does that make sense? And the structures of the system that we're going to be learning about are going to be primarily related to ventilation. So most of the anatomy you guys are going to be is about how we conduct air. The anatomy of tissue gas exchange you guys have already learned. Capillaries are really, really thin. They have basically just a single endothelial layer. You know, that's a simple squamous epithelium with a basement membrane. So we already have a pretty good idea how this occurs due to the anatomy of the system. Transportation, we've already learned about, or been taught about. where we're talking about hemoglobin and hemes, and we'll end up actually talking about that a little bit more too. Alveolar gas exchange, we already know Fick's law, but we are definitely going to talk more about the anatomy of our alveoli and how they promote respiration and what would happen if we start to impact factors like water in your lungs. All right, does everybody have just a relatively decent grasp of what we're going to even be doing with this system? When you guys go to study at the very end of this unit, 2 weeks away already, this is a great reference chart, sheet, whatever the word is for this, for what you will need to study. You will need to know all the physiology of steps 1, 2, 3, and 4. Okay? Make sense? Cool. Let's take a little bit of time to go through some anatomy. Now, what we're going to start off with here this semester is just the lecture. And when we make it to the lab today, today's lab is on respiratory anatomy. So when we make it to the lab today, all the models, pictures of models that we have are related to ventilation, except for one where we're going to have an alveolar sac. So you're going to start to see that respiration occurs down there. If you look in the lecture, you'll see my very first slide is exactly what we just went through. There are 4 processes of respiration. First and foremost is pulmonary ventilation. What equation do we know will help us understand pulmonary ventilation better? Because it's all about airflow. Good. Pressure gradient divided by resistance. Even if you didn't write that down, hopefully that made sense to y'all. Now, do you guys think those are the only things that are involved in pulmonary ventilation? I hope the answer is no. There's going to be some other things that are involved in pulmonary ventilation that I was discussing in that introduction, but we'll be learning about those because all of the anatomy of this system relates back to this. And there are other functions even of the respiratory system. We're going to be focusing a lot on the respiratory system as a passageway for air. We definitely will. A lot of our physiology is going to be dealing with this. Gas exchange. Yeah, most people think that this is the only function of the respiratory system, but it's not. Ventilation and respiration. Bullet point 1 is ventilation. Bullet point 2 is respiration. Sense of smell. Since we breathe through our nose, as we create this vacuum that sucks the air in from the world around us, it brings all that air into our nasal cavity. And what do we know? What portion of our brain communicates with our nasal cavity right here through the ethmoid bone? What nerves lie down here? You guys are like, that's AMP1. I hate you. Yeah, thank you, Parker. This is the olfactory, right? Olfactory nerve rootlets, the olfactory bulb right above. Yes, no, maybe so? Well, one way or the other. We won't talk as much about olfaction. That should have kind of been a part of AMP1 near the end of your semester. Should have kind of parta. But those little nerve rootlets, they actually sit in that endothelial tissue of the nasal cavity. They're receptors. The ends of those olfactory nerve rootlets are receptors. And as chemicals enter your nasal cavity, those chemicals dissolve in mucus. This is the only time I'm going to be talking about this, so you might want to write it down. This could be like a homework problem. But your nasal cavity is filled with mucus, filled, covered, right? Hopefully not filled, because then you got a cold, but you know what I mean. It's lined. Your nasal cavity is moist all the time due to the mucus being produced by the sinuses. And as chemicals bind to that mucus, it dissolves. And as it dissolves, then it interacts with the receptors. If it does not dissolve, then it cannot interact with your receptors. I'll repeat, if a chemical does not dissolve, then it can't interact with those receptors. Chemicals also have to diffuse through the fluid to get to those receptors. So what do we know about the rate of diffusion? It's inversely proportional to the distance it has to travel. So do you breathe better or do you breathe worse? Do you smell better? Sorry, do you smell better or do you smell worse when you got a cold and your nasal cavity is just filled with phlegm? And that's because the chemicals, you're not getting them into your nose as well because the air passageway is blocked, and they have a ton of direction. They have a lot of distance that they have to travel, so you're not going to be able to smell very well. What about if you have a dry nose? Melissa, what is Arizona like, right? Who's ever been to a super dry desert? And you're out, yeah, it's super dry. I mean, basically 0% humidity, and it's 115 degrees out, and your nasal cavity starts to dry out, and you're going to start to not be able to smell very well pretty quick. Yeah, Death Valley is super cool. What a beautiful, beautiful desert. Tucson's still my favorite desert in the world, though. Verdant. It's the greenest desert you'll ever be. Palo Verde, all the trees are green, for Pete's sake. No leaves. The trees are green. The bark is green. The bark has chlorophyll. Crazy. So cool. Anywho, speech. Again, this is going to be pretty much the only place we talk about speech itself. We will talk about the larynx, and we'll be talking about some of the anatomy of the larynx that allows for speech. But I want everybody just, to say something. But this time, for probably the first time in your life, focus on your diaphragm as you speak. What's happening to your diaphragm as you are speaking or saying something? What are you doing to your diaphragm? Can you guys feel, you guys being able to focus on it? I like it, Andy. Flexing. You're contracting your diaphragm, right? Everybody sing something. You should be able to feel that diaphragm contracting. What direction is air going? Is it going into your lungs or is it coming out from your lungs towards your mouth? And that's important. The way that our anatomy is set up, it's not designed for speaking while you are breathing in. Everybody try to say something. Many people have never even tried this before. It cracks me up. Try to speak or sing while breathing in. Just give it a try. Other people in your house might think there's a devil possessing your body. Right? It's just bizarre. True story? Anybody else making themselves laugh trying that? Hello. Some people can't even do it at all. The role of the respiratory system in speech is pretty darn critical. If you're not using your respiratory system properly, speech becomes extremely difficult. Okay. And there's some muscles involved in that, and we'll kind of look a little bit at that larynx and some of the cartilage and muscles involved in speech, but we won't focus on it too much. Other functions. Please write this down right now in bold, huge capital letters, underlined and asterisks, and highlight it, and call your tattoo parlor, and you will be tested on this, you will be tested on this, you will be tested on this. The respiratory system controls 70% of all your acid-base balance. 70% of all of your acid-base balance. 70%. It's by far the most important system for controlling your acid-base balance. Acids are bad. Bases are bad, but acids are really, really bad, and we're fighting acid all the time. I'm going to be teaching you guys what's going on with the respiratory system and acid, what the link is. By the way, you guys have already started the link. You just may or may not know that you've started that link. But who's ever had ceviche? It's like one of my favorite foods on the face of the planet. Ceviche. Yes, no? Some people are like, man, yeah, no. Ceviche is, some people think it's raw fish or raw seafood, but it's not raw, right? So if you take, little shrimpies, pieces of fish, you cook it in lime. You take lime acid and you put it on the fish. And fish is such a delicate meat that it can cook thoroughly in just a couple of minutes in acid. So acid is the cooker. It actually does something called denatures proteins, and we're going to walk through that when we get into the digestive system. We'll actually talk about it later in the respiratory system too. Acids will cause your proteins to unfold. That's why we have hydrochloric acid inside of our stomach, is because it's there to help break proteins down. Acid-base balance is about acids in your bloodstream. Do you have proteins in your bloodstream too? Trying to live their life, do their thing? You started to learn how important those blood proteins are to daily function, whether it's blood clotting or whether it's blood pressure. Yes, no, maybe so, not to mention all the other myriad of things that all your blood proteins are doing. And so if acid level starts to increase inside your bloodstream, shit, now we're starting to cook those proteins, literally cooking the proteins in your blood. And it doesn't take a very big change for your blood proteins to start to get cooked. It takes a very small relative change for your proteins in your blood to start getting cooked. All you have to do is exercise. Exercise is still one of the best things you could do to your body. Don't get me wrong, okay? Exercise will start to cook your blood, though, and then your body's going to fight it. Because what happens to your respiratory system as you start to exercise? Do you breathe faster, slower, bigger, smaller? This is your body fighting against acids. It's not your body getting more oxygen. This is one of the most common misconceptions. Does your body get more oxygen as it breathes faster? Of course. But your body is not breathing faster during exercise primarily to get oxygen. It's to get rid of CO2 and that's going to get rid of acid. So we'll talk a lot more about that. 70% of your acid-base balance is controlled by which system? Which system of your body controls 70% of your acid-base balance? Good. The other 30%, by the way, is controlled by your kidneys. ACE, both ACE1 and ACE2, angiotensin converting enzyme. That's pretty darn important, right? Blood pressure. So now we're dealing with an organ that not only helps us breathe, but it also helps us with proper blood pressure and proper acid-base balance. Gulp. It's pretty darn important organs of your body. Venous return. Everybody take a nice deep breath. What just happened to the volume of your thoracic cavity? Increase, decrease, no change. And as those lungs expand inside of that thoracic cavity, all of the pulmonary circuit that's in there, all those blood vessels, have an organ that's squeezing on them. And the blood that was sitting in all of those pulmonary vessels is going to go squirting right back to the heart. Does that make sense? Because there's pressure on those blood vessels and they don't have any way, they can go backwards. So as you start to squeeze those blood vessels in that nice deep breath, that blood's going to go forward. Just like if you're squeezing on your vena cava, the blood goes to the right atrium faster, but now you just took a nice deep breath in, and all that blood that was pooled in your pulmonary circuit is going to rush back to your heart, to the left side of your heart. Boom, boom. And then that heart beats, and all of that extra blood rushes out to the system. So everybody, take a deep breath in and focus on your head. This doesn't work for everybody, but give it a try. Focus on how your head feels. Did you guys kind of feel just it's hard to describe. Do you guys feel a little bit more energy? Yeah, like your eyes actually kind of work a little bit better. It's a real funny thing to try to describe to students, but try this. Give it a try. Some of you guys are saying yes, and some of you guys are like, WTF, Dr. H, I don't know what the hell you're talking about. Give it a try, especially when you're not physically like just focused completely on it. Like while you're doing something else, take a nice deep breath and focus on how you feel. And I don't know, I'm watching a lot of Daniel Tiger because my kid is 4 years old and I'm trying to teach him to deal with his emotions. What do they tell you to do when you're 4, when you're angry, right? What are you supposed to do when you're angry? Take a deep breath and count to 4. Yeah, that's right. I got it. You're supposed to take that deep breath, and what it does is it's going to trigger this exact reaction. When you take that deep breath, blood flow to the heart increases, and then that heart beats, and now more blood gets out to the rest of your systems, and the number one system that receives your blood is your brain. Does that make sense? So now your brain gets a shot of extra blood. It gets a shot of extra oxygen and glucose. Doesn't that sound pretty damn good? Especially if you start getting angry and your blood vessels to your brain are vasoconstricting, now all of a sudden you get more blood out to your brain. Yes, it helps everything get better. This is what meditation is, in case you guys hadn't made that connection yet. Meditation, breathing techniques are all about slowing portions of your body down that don't need the oxygen and getting oxygen to the areas of your body that do need it. Primarily your brain. It helps your brain heal. And there's a lot of physiology behind it. If anybody's ever had anxiety attacks, and if anybody's ever gone to see a doctor or a psychologist about it, one of the first things they'll do is teach you how to breathe. If you've never ever looked for treatment for your anxiety, there it is. One of the first things you can do is learn how to breathe. I'm not talking about, Dr. H, I know how to breathe, man. I've been doing that my whole life. No, that's not what I'm saying. Right? I'm saying there are breathing techniques that allow you to get more air to your brain. And I've had lots of students through the years that actually pick up those breathing techniques and are finally, for the first time in their lives, able to control their anxiety. And it's all about venous return. All right, one more time. Take that deep breath. You just breathed in 1 million bacteria. I already did this one earlier, but we better have a pretty good immune system in place. We don't want that bacteria down in our lungs. We don't want that virus down into our lungs. We're going to try to get rid of it before it gets there. Yes, no, maybe so? This is why you can even be exposed to low levels of COVID, even at the beginning of the epidemic, low levels, low small particles per million, and our system should have been able to filter it out. But COVID, unfortunately, our body doesn't have as many ways to be able to filter it out because we don't have an immune system function against it. So when the parts per million got just a little bit too high, that's when it gets down into the lungs. Makes it down to our ACE2 receptors and uses those ACE2 receptors to get into our blood. Damn you, COVID. All right, so in the lab today, this is what we're going to be focusing on. We're going to be focusing on this air passageway, and we're going to break it down into primarily just one flow chart. But I want to teach you guys that these, like this figure right here, it shows two different types of organization, two different subclassifications of organization. One is considered structural, and the other one is considered functional. You know me, I like function, so I tend to focus on functional organization. And I recommend you guys do the same, except for one little itty tiny little bitty detail. It's probably a lot more than a tiny detail. Structural organization is where we get the terms upper respiratory tract infection, lower respiratory tract infection. You guys heard these terms before, especially if you've been working in a hospital, or if you've had kids, or maybe you've had your own. Upper respiratory tract infection is just from the larynx up. So if a doctor is talking about an upper respiratory tract infection, they're not talking about the trachea, they're not talking about the bronchioles, and they're definitely not talking about the lungs. They're only talking about what's happening from the neck up. So this would be like an infection at the back of the throat, some sort of pharyngeal infection, nasal cavity infection. Whereas a lower respiratory tract infection, this is what we want to prevent. So this is what my daughter had. By the way, you guys want to be interested, my daughter had a virus. So remember, she was sick basically all last week and over the last weekend. She had a virus. She's still wheezy, but she's better. You guys want to just have, if you like viruses and weird respiratory viruses, she had Boca virus. And I'd never heard of it. 43 years old and 20 plus years of that just studying science and medicine, and I never heard of the Boca virus before. Anyways, the Boca virus is a lower respiratory tract infection. It's a virus that gets down in the lower respiratory system, primarily of kids that are less than 2 years old. And there's my daughter, right? She's right in that age category. And it causes a lower respiratory tract infection. So she started getting this viral buildup and lung inner fluid in her lungs due to it. Thank you, Beck. She'll be okay. She's positive energy and stuff right now. Just still a little wheezy. So we're trying to get it figured out. But functional organization is going to be the easiest way for you guys to learn the anatomy, in my opinion. How so? I'm going to do something a little different here. Can't say I do this very often, but I just had an idea. Oh, come on, program. Make this possible for me. So I don't do this very often where I divide up the screens, but I think this is actually going to be pretty darn helpful. So everybody take a deep breath in. And what's the first part of your respiratory system that air is going? Where's it going first? I know we did this earlier, but just work with me here. So it goes through the nares first. Good, good. But where does yeah, no, sorry, Heather, that's a little bit of a trick on my part because I did talk specifically about nares earlier. But this time around, I'm looking for what general area it goes first. Sorry, Heather, I didn't make that clear. So it's going into the nose and it's going into what's called the nasal cavity. Does that make sense? So I can get everybody started on this. So the very first place that this air goes is the nasal cavity. Okay, it's going to enter through a nare cavity. It's going to go through a nare. It's going to enter the nasal cavity. And there's some anatomy going on inside this nasal cavity, but let's ignore it right now. Because what we do is create a simplified flow chart that helps us start to understand the anatomy a little bit better. And then we're going to start to take this flow chart and we're going to expand on it. So let's start simple. Okay, the nose is just the upper nose portion. And we can actually write some anatomy in for the nose as well. Goodness gracious, where are we here? Because the nose is exactly, you touch your nose, right? That's not the entire nasal cavity. So let's go ahead and throw that in. Nose to the nasal cavity. I tend not to test on the nose. I tend to focus more on the nasal cavity, but we don't want to skip too much here. Now, that air is going to flow through the nasal cavity, and you can see that in this picture right here. Okay, and it's got these weird curves. If you recall from A&P1, these are part of the ethmoid bone up above, the superior and middle nasal concha. And then there's a single bone down here called the inferior nasal concha. Inferior is the biggest. It's again one of those weird slips. Most students think that inferior means little, but it doesn't. It means the most low. These have a function. We'll talk about them in a little bit, but they're curved like a shell. And if you're going to flow air past something, if you check out my hand, if you're going to flow air through something that's curved, like so, it's going to start creating a vortex. Does that make sense? That if I have a shape like this, it's going to start causing that air to almost turn into a tornado. Yes, no, maybe so? It's like a wave that somebody's surfing on, right? So yeah, nasal cavity is going to be pretty darn cool. We're not going to focus on it too much, but we definitely will focus on it. But from the nasal cavity, it goes to the throat. Now the throat starts way up here by the ear. And most people don't understand, they haven't learned that before. Let's go that route. Not don't understand that, they just haven't learned that before. But way up here at the tip of my pointer, this is the throat. And it runs all the way down to our voice box, the larynx. All of that is our throat. And in medicine, we call the throat the pharynx. Now, the pharynx is going to be divided into 3 different regions, and we're going to write those into our flow chart in just a little bit. But again, we want to start with something that's a little bit more simplified. From the pharynx, air now is finally going to pass with the help of a nice little cartilage flap right here, we're going to talk about, called the epiglottis. Air should flow down into the larynx. The larynx is our voice box. And the larynx needs a little bit of recognition for its anatomy. Its anatomy is extremely unique. It's completely made up of cartilage. And you guys are going to be learning a whole bunch of different pieces of that cartilage that help make up the larynx, and some of the membrane flaps and stuff like that that help us with speaking. So we'll come back to that. From there, all of that air passes down into what we know of as the windpipe. Most people have heard that term, windpipe, maybe? Yes, no, maybe so? But we're not learning the windpipe. Don't call this a windpipe on the lecture or the lab, of course. What we want to call it is the trachea. You may have heard of, a tracheotomy or something like that before. Okay, that's helping a patient breathe, where you cut the between the third and the fourth cartilage of the tracheal rings. The trachea is completely surrounded by these C-shaped tracheal cartilages, and there's this tissue in between that you can cut through. And if you need to help a patient breathe, sometimes you have to get past the upper respiratory area. We need to get immediately into the lower respiratory area, and that's where they do tracheotomies. So they can jump right down into the trachea, throw a tube right down there, and that's going to help that patient get ventilated. So now we're finally down into the lung itself. And in the lung itself, this huge trachea is going to branch. And then right where it branches, you know, we can draw in a branch if you want. I'm not going to draw in the branch because it's a mirror image on either side, and I think it just kind of ends up taking up room on my flow chart. But sometimes it's nice to draw in anyway. So you guys get to make your own decisions on how you want to study. But the trachea is going to branch, and it's going to become the bronchus. But what I don't want to do right now is get you guys on the wrong start with the bronchus. Bronchus is singular, but the bronchus is going to go, and you can look at it in the picture. Can you see that the bronchus is this portion right here and this portion over here? And what you'll notice about the bronchus is that inside the lungs, it's the largest of the tubes, but the bronchus is going to branch. And that branch is going to branch and branch and branch and branch, and it's going to continue to branch. And every time it branches, it's going to get smaller and smaller and smaller and smaller. Yes, no, maybe so? Nice. Each one of those branches has a name, and there's 12 different orders of branching. I'm not going to make you guys learn all 12 different orders of branching. No way, no how. So we're just going to start with what's the first branch of the bronchus is just called primary. Okay, it's the primary bronchus. It's the biggest of all the bronchi. Bronchi is plural, bronchus is singular. The primary bronchi split. Now you guys can probably guess they become secondary bronchi. And then those branch. And what are those going to be called? Tertiary. And then it goes quaternary and whatever, cinquenary, sessionary. I don't really know all the different orders, and I never really tried to memorize them. There are lots of different branches of these bronchi, but each time it branches, it's going to get smaller and smaller and smaller until it becomes so small that it's going to start to really lose all of its cartilage. We're going to end up talking about the epithelial layers here. We're going to talk about where cartilage is found and where cartilage is not found. So you're going to see you're going to add a lot to this flow chart. We just need something to start with. But as it branches, many different orders of branching, it's finally going to get really small. And what do we call small blood vessels? It goes artery, and as those branch and get smaller and smaller and smaller, what did they become? It's going to be, the bronchi will become, yeah, arterioles, right? Is everybody on board with me? Just trying to teach you guys a way to easily remember this. Yes, no, maybe so? For those that aren't, you're all right. I don't type well either. Arteries became arterioles. And what was so unique about arterioles? They're small. They led out to the capillaries, but what else was really unique about the arterioles? Resistance, good. Circular bands of what? Great. I love that you guys are coming with these two key words. Amazing. Circular bands of smooth muscle. Yes, yes, yes. And therefore, we are able to change the diameter of those arterioles. Boom. You guys just learned something about the bronchioles. They're the exact same as the arterioles. Okay, so as we get out to that 12th order of bronchi, smaller and smaller and smaller, and we're losing cartilage, and we're actually getting thinner endothelial walls, these guys start to get surrounded with smooth muscle. And when they do, they become what's called terminal bronchioles. Terminal bronchioles are the end of the conducting zone. They terminate the conducting zone. That's why they're called terminal. So everything that we've talked about so far, all of this, everything here, is all part of the conducting zone. Everything helps us move air or airflow. Yes, no, maybe so? It's all about conduction so far. But now we're done conducting. Now we're going to start having gas exchange. And what word did we learn for gas exchange not very long ago? What word is used? Respiration. So everything from here on out is going to be part of the respiratory zone. Where gas exchange occurs is all part of the respiratory zone. And this all occurs down in a structure better known as an alveolar sac. So if you look at the picture right here, up at the very top, this is a terminal bronchiole in this picture. And the terminal bronchiole, yeah, the grapes, you got it. The terminal bronchiole, that divides. We're not done branching. Just because we call it the terminal bronchiole doesn't mean that it's the end of the road. Such a pain. And this terminal bronchiole is going to continue to branch, but where it branches, it's going to start to lose the smooth muscle even. And gas exchange is going to start. So we're not going to be calling it terminal bronchioles anymore. We're now going to be calling them respiratory bronchioles. They're still not alveoli. They're leading out to the alveoli. So not a lot of gas exchange will occur here. Finally, these bronchioles are going to completely end, and they end in these pore systems. That is very difficult to see with what we have right now. Decent resolution here. This that my mouse is over right now, this is a respiratory bronchiole, and it's going to branch, of course, but it branches into just a pore system, these open sections of the tube. They literally open up and they lead out to each individual alveoli. Okay, this open space is called an alveolar duct. The alveolar duct is what finally gets the air out to each grape, which is the alveolus. Alveoli is plural, alveolus is singular. So we're going to go alveolar duct. And what makes the alveolar duct different than the respiratory bronchiole? If I have a different name, then I have a different structure. And if I have a different structure, then I have a different function. Does that make sense, everybody? What makes this unique is the alveolar duct is where we start to get simple squamous epithelial tissue. So what did that do to the distance that air has to travel? And therefore, what happens to the rate of diffusion? Increase, decrease, no change. So when we make it to the alveolar duct, that's when diffusion starts to just act as a torrent. Torrent? Torrent. There, that's the better word. Torrent is like something that you could shoot weapons off of or something. I can't remember exactly what a turn is, but torrent. And that air is going to pass down into the alveoli, and here inside these alveoli is where 90+% of all of your gas exchange occurs. Okay? Can't get this up any higher. Started out with my arrows a little bit too big. I do that every semester. It's funny. So what we're going to do after our break is that we're going to start to look at your objectives. They are simple squamous. Yes, Melissa, they are. I don't know where I went wrong there. Alveolar ducts are simple squamous, and alveoli are simple squamous. But an alveolar duct is simple squamous that's a tube, whereas an alveoli, or as an alveolus, singular, is a circle. Okay, now we got a pocket of simple squamous epithelial tissue. So structure determines function. Alveolus is a circle. Alveolar ducts are tubes. Yes, no, maybe so? Cool. Yeah, so what we'll do when we get back from break is that we're going to start to talk about what is some of the anatomy of the nose and how does that help with the function of the respiratory system. What's some of the anatomy of the nasal cavity, anatomy of the pharynx, anatomy of the larynx, trachea, bronchi. Yeah, there's some things to learn about the bronchi. Bronchioles, we'll definitely focus more on different types of bronchioles, terminal bronchioles. Same idea out here, bronchial, terminal bronchial, and then what's happening out here in the respiratory. Yep, so let's take a break. Let's take a, I don't know, 19-minute break. So everybody be back at 45 after, please. Okay, welcome back. And let's get rolling. Hi. Let's get rolling. Let's get rolling. So lab material will coincide with lecture material because lab material here for Unit 3, the very first one, is going to be anatomy. Lots of cool anatomy stuff. Let me, I meant to do something. Those are docx. So lots of things that I have already, by the way. So I'll go ahead and just open this one up for you so you guys can see this. This is actually just a little worksheet. It's not due. There's no grade associated with it. It's just an additional study guide for you all. So you can label and structure the conducting zone. So just stick with the conducting zone. This is going to be the upper respiratory conducting zone. So you're going to go find your objectives, which are right there on Blackboard, and you're just label stuff, okay? Sound good? Just label stuff. Don't overthink this. Just label it. External nasal nares, nasal cavity in the nose, concha, which are going to create meatus. We'll go through each one of those. Pharynx, back of the throat, nasopharynx, oropharynx, laryngopharynx. So we'll go through each one of those, and then you guys can go through and label it. Same thing going on here, right? This is a terminal bronchial right there. So this is a respiratory bronchial. So what are all the rest of these things going to be? Lots of them are going to be alveolar ducts, respiratory bronchioles, and alveoli and alveolar sacs. So you guys can go through, label that, label the following: trachea, right, left, right. All of this sort of stuff tells you basically follow your objectives, label these things. There's also going to be a thoracic cavity labeling thing for you guys to go through because the anatomy of thoracic cavity is critical. This actually is not a bad time to talk about this. We'll come back and talk about this. We have to remember the same thing that we had to remember for the heart, and that was the serous membranes. There's serous membrane around the heart, serous membrane around your lungs, and a serous membrane all around your abdominal organs. And we have to remember these serous membranes. They're a critical part of a lot of different body functions. And without these serous membranes, you wouldn't breathe. They're really that important. One of the most important anatomical features of the respiratory system has to do with these pleura and the pleural cavity, parietal versus visceral pleura. You guys kind of remember those names-ish? Love that Andy just got yes right off the bat. Got some more yeses now. If not, just do me a favor. Even if you did and even if you didn't, make sure you highlight or write these three things down: visceral pleura, parietal pleura, pleural cavity, and highlight and asterisk those. You're gonna have to be able to identify them. And you're gonna have to tie them in with the physiology. Just promise you guys straight up, you will be tested on that. Right there, I have them highlighted. Write down visceral pleura, parietal pleura, pleural cavity. You will be tested on those. So understand both their anatomy, and we will get to their function. We're going to talk about it in detail tomorrow. I also stole something from another professor. Pretty good little thief. I borrowed it with no intention of paying them back for it, but I will give them credit for it. They took the objectives and they did some pretty awesome stuff with it. So they basically, threw some videos in here. These are not my YouTube videos. These are somebody else's YouTube videos. But I'm going to be showing you guys that you have access to my YouTube videos too now. Finally, I start doing my own anatomy YouTube videos for the respiratory system. There's a basic introduction for the function of the respiratory system. We already did that in lecture together. But what they do is they go through each one of the objectives here in bold, 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 Koana, like Moana, right? Pretty fun stuff. Vestibule, nasal cavity, superior nasal meatus, middle nasal meatus, and inferior nasal meatus. But they don't just, bold it, which a lot of my objectives are just, hey, here's the word. But they actually went in and they kind of defined the general area where to find these things. This is both a blessing and a curse. I think a major learning technique for me personally is to get dirty, which means I need to go and find these things for myself. If I just look at pictures, if I just look at words that describe where things are, that's not enough for me and my learning. Does that make sense? Like I have to go find an objective word and I have to go find it myself. It's like part of my personality too. I'm real type A, as you guys can probably tell. I'm like, I got to do it. I'm going to do it myself. I'm going to kick and I'm going to scream and I'm going to make sure that I don't rely on somebody else too much, because then I do a little bit less learning. That's for me. Other people need to lean on other people for learning. We all have our own different ways of learning. So if you like this sort of, here's a descriptor, find this area of the nasal cavity inside that nose, boom, it's there for you. And then you can check it off once you find it, which is pretty cool. So anyways, I found this and went, man, I think some of my students would really like this. And so I stole it, but I haven't really found too many of these since. But these are your objectives. So that's one way to go through and look at your objectives for today's lab. Why did that pop open? Respiratory models. Okay, we'll just open up the PDF here. Here's the version of the objectives that I give, right? It's just a list. Nose, pharynx, larynx, trachea, going down into the bronchia, down into those alveoli, the different pleura, visceral, parietal, and the pleural cavity, which is what I just asked you guys to highlight. Then we're going to go in and we're going to talk about the lungs. And then, of course, because you guys, you call for it every semester. This is your favorite thing to do. It's histology. And, I just can't let my students down, since you guys every single semester write me how much you love your histo. I'm not being sarcastic at all right now. We still got to go through our histo. Okay, so that's what we're going to do to get together. Which should actually, for the first time this semester, maybe be done a little bit early today. You guys ready to get rolling? Oh, I guess one more thing. You guys like, yes, I'm definitely ready to get rolling. Is that I have a doc that you guys can open up. This is the last thing. It says YouTube videos by Dr. H. So these are the links to my YouTube videos, or if you already know where my YouTube HTML, I don't know. I don't know. I'm not tech savvy. My YouTube stuff is at, you guys could already find. But these are the models that I have to test you guys on. I have a half head model. I call it the hemi head. I got a larynx model. I got a bronchial tree. I got a thoracic organs. It's all, basically it's a model of just the thoracic organs. And then I got this big old alveolar sac model. I also have big torso models, but you can find all of these different structures on those as well. So this is a great thing for you guys to be able to just, control click, should open up something that ends up being just me in the model. These guys are not extremely long, so these are not going to be a big waste of your time. Is this really a 3-minute ad? Now, I tried to avoid anyways, you can see there's a little bit of shake sometimes, but But anyways, I just go through this Hemi head model and I literally walk you guys through the nairs and each one of your objectives. And I'll have a pointer and I point it at each part. So nice. Channel. Thank you, Heather. I didn't even look. Anyways, I can't stand the sound of my own voice, so I'm sure you guys are the same way. I can't stand the sound of his voice. Okay, and then the one last thing, of course, is you guys already are well aware that you get extra credit for anatomy days. Uh-huh. You're well aware. True story? So here's pictures. Pictures, pictures, pictures. Lots of pictures. Got my big nose model. I got, you know, some things already potentially labeled for you, but you can get rid of those labels, just move them around. I got my tracheal, my respiratory tree model. This is just a picture, zoom in of the larynx, different portions of the larynx we'll talk about. Here's the cartilaginous laryngeal model right here, and you know, just different parts of the lungs. So quite a few different pictures of things for you guys to walk through and label. Let's do this. Give me 37.5 seconds. Trying to look through my files real quick. What just happened? No, I don't want that. I want, all right, make it 47.5 seconds. I clicked on the wrong button. How many seconds do I have left? Aha, I do got one. Okay. Good news is we have a drag and drop. And I will get that. Actually, I can do that right now. You guys use any of that drag and drop from last system? Like for blood vessels, for it was kind of limited on what you had. This one is going to even be even better because this is going to be the entire system. This one's going to be great. It's great. All right, so submit that, and then I'll open this for you guys. So now you guys got a drag and drop, and we can actually use the drag and drop. It's pretty powerful. Oh, please don't tell me I just accidentally closed it. All right, here we go. We're going to use it. I did not. Okay, we're going to use this drag and drop. It's again created by a professor at ACC, Joan Hauser. This particular one. We're going to use this drag and drop to start actually walking our way through the different models. And we're going to be talking about some of the physiology of each one of these structures as well as we go through. So the first place that we're going to look is what's happening. Ha, that's what I'm for, Heather. Don't be shy. I got a few people. You're not normally shy with contacting me. I got a few people that aren't normally shy. If you are looking for, you know, correct answers, that's what I'm here for. I want that, y'all. Stop thinking that you're, you know, pestering a professor by asking for help. Isn't that my job? Isn't that what I get paid for? Isn't that what I love doing? So I'm not just going to give you guys answers. Let me spiel on this just for a second. Not according. Oh yeah, some professor. What? Who cares? Who cares about them? You're with me. Listen. Yeah, I mean, it is what it is there. Um, the easy route for me would be to give you guys a key to everything. That's easy. And some students think that they'd like that route. Why? Because it's easier for a student as well. Again, I was a student for 17 years, and when I had access to the answers, did I do all of the work required to double check, triple check my own answers? Hell no, I didn't. I didn't even come close. I'd go, oh, I think it's this answer, but I'm just going to check my answer key. Oh, yeah, I was right. But I didn't have to think about why I was right. Or if I was wrong, I'm like, oh, crap, I got to change that. But I didn't have to think about why my answer was right. I didn't have to think about why my answer was wrong. So work your way through each one of these, do your best, but then reach out to me. I'm the answer key. Does that make sense? Yeah, it's always the path of least resistance. What really bugged me, you guys remember, I don't know whose teachers did this, but you'd have homework problems out of the textbook, but only the odds. You had like the answer key in the back of the book to the odd questions, but then of course the teacher would only give you the evens. It's like, son of a, ugh, how am I supposed to check? Yeah, super old school. I don't know if they still do that. All right, enough on that spiel because I don't seem to be getting a lot of feedback from you guys on it, but it is what it is. That's my style, and again, you guys have me as a professor, so you got to deal with my style. Breathe in. All of that air, first and foremost, passes through the nostril. Now, the nostril is not called the nostril. Of course not. That would be just too easy. It's called a nair. And a nair is an opening. There is an external nair, and you're going to find that there's also an internal nair called the choana. But we can call this an external nair / nostril. So on a lab practical, if I'm pointing to the opening, if I'm pointing to the that's not even the nair right there. I would find a picture that right here, that's a good one. See how this is tilted to the side so I can actually look at the opening of that nostril? That right there is an external nair. Does that make sense? External nair. I feel like something's missing from here, but that's okay. Oh, I guess not. Now, as that air enters the nose, the first thing that's going to end up happening is it's going to kind of hit this area that's highlighted in yellow. And this area that's highlighted in yellow is kind of like a welcoming area for air. It's this kind of curved region on this model right here. You can see the three-dimensionality of that region of the nose. This area right here is called the nasal vestibule. Do you guys notice how I'm not just calling it vestibule? We're calling it the nasal vestibule. Yes, no, maybe so? Even though on your objectives list, you're going to see it just says vestibule. Just says vestibule. But look, it's all in a subset of nose. Nose is nasal. So nasal external nares, nasal internal nares, nasal vestibule, nasal cavity. Do you think there's other vestibules in the body? Well, the answer is yes. Many other vestibules, whether you know that or not. So we're going to be learning about other vestibules. So if it is a name like vestibule, like medulla, like cortex, you better be telling me what vestibule it is. The vestibule is not an opening, Heather. The vestibule is actually a roof. Vestibule in Latin means covering. So the nasal vestibule is the area that air will be passing through. Here's the context that makes it easier. The nasal vestibule is where hair grows from. So your nose hair is growing from the nasal vestibule. Unless you got it lasered out, and then it's not growing from anywhere. First line of defense, peeps. First line of defense is that nose hair, but most of us trim it away. Now, all that air is going to pass up into this big open space. The open space is just the nasal cavity. All of that open space. By the way, in this pic, nasal vestibule is this arrow pointing to that three-dimensional darker area right there. And as we come across it on other pictures, like we already looked at this picture right here, I'll label that for you as well. Is everybody pretty clear on external nares and nasal vestibule? And as it enters this chamber, the chamber, all here in purple, is called a nasal cavity. The nasal cavity is all of that open space. Cavity, open space. But that open space has these concha. Concha means shell in Latin on the inside, right? Actually, we're done with this drag and drop right here. But you can see it really nicely in this picture, and probably even more nicely in this picture. You take away that nasal septum, that's exactly what's dividing the right and left nasal cavities from one another, and you take away all the membrane. What you end up seeing are these structures here. You guys see these that kind of look like sponges? These are bones. Well, I don't know whether you saw this model in your A&P1 class or not. This is my big nose model. I love my big nose model. But some students don't end up seeing this. But this right here, if you take away all the membrane, these are bones. And what you see up here that I'm outlining with my mouse, that's your ethmoid bone. And that ethmoid bone lies all throughout that superior aspect of the nasal cavity. This shark's fin up top right here, that's the crista galli. Crista galli, if you guys remember learning that structure, that's where the dura mater of the brain attaches to the ethmoid. Crista galli means rooster's comb in Latin. Anybody know Spanish, right? Gallo is rooster. Crista galli means rooster's comb because it kind of looks like the crest of a rooster's comb. And then everything below is all part of that ethmoid. Now, big portion of those ethmoid features were the superior nasal concha and the middle nasal concha. And the inferior nasal concha was a bone all on its own. So that bone down there is the inferior nasal concha. And what separates the oral cavity from the nasal cavity is a bony structure. Boom, right there, called the hard palate. Back behind this would be the soft palate, the hard palate, or the palatine bone. It's a really important key bony structure that's going to separate the nasal cavity from the oral cavity down below. Concha, though. Concha. The concha themselves are the bones. They're the curves. But where that bone meets the rest of the nasal cavity, there's a groove, right? Emperor's New Groove? There's a groove. Get your groove on. It doesn't, I mean, I guess it technically means the air is going to start dancing. Because this groove is the vortex. As air passes through this, it's not just a rush of air into the nasal cavity anymore. It hits this bone and it starts to swirl, like really high speed, super high velocity vortex. And your nasal cavity is lined with mucus. Your entire nasal cavity is moist. It's got mucus that lines and coats everything. And so as the air swirls through these grooves, it's going to slam that air down into the mucus. and hopefully help with it dissolving any sort of particulate matter that might have gotten through your respiratory nasal vestibule. So now if we get bacteria or if we get dust in the air, the idea is we want to slam those particulates in the air, we want to slam them down into our mucus as best as we can. And then that mucus is actually going to drain down the throat and hopefully go into our digestive system. And then we just swallow that bacteria, swallow that stuff. Or we can do, a farmer's rocket. You guys know what I'm talking about? Just plug one nostril and blow it out real good. That's always, that one's always fun. A streamline in the air isn't going to slam the air. The vortex causes it to swirl and it increases the speed at which air is flowing, and therefore it creates this curl. I can't really show you very well. I mean, imagine air passing in like this, and it swirls as it's actually going to go this direction on this model. It's going to swirl and slam down on the mucus rather than just pass by the mucus. That's the benefit. Yes, no? It's not a vortex rushing in this direction. It's a vortex rushing from superior, boom, and it slams down on the mucus below. So again, now we're looking at this model. Remember, the superior nasal meatus is stupidly tiny. It's tiny, even though it's called superior. The biggest one is inferior. That's created from its own bone. The middle nasal meatus is created from the ethmoid bone. The nasal vestibule, that air is entering, and that's where the hair would grow. Done a pretty good job labeling these first few so far. Hard palate, bone. Soft palate, the very end of the soft palate back here is called the uvula. Uvula we're not going to be really dealing with much until we get to the digestive system. It's a pretty important aspect of the oral cavity. But it's also kind of cool to think about with the respiratory system. So the uvula is like a soft flap that as you swallow food is going to put pressure on this soft flap, like a valve of the heart. And so the pressure is greater on this side than it is on this side. Then it closes that valve. Does that make sense? Just like the valves of the heart. Yes, no, maybe so. If the pressure is greater down here than it is up here, then that uvula closes, which is great, because do you want food shooting out your nose? Depends on who's bet you to do what. But now eat or drink especially, some food, and then somebody tells you a penis joke. Now all of a sudden you giggle, and when you giggle, the muscles of this around this area are going to vibrate, and the uvula is going to move back and forth, right? And if you're in the middle of drinking a Pepsi when that joke happened, that's when you get the milk, the Pepsi shooting out your nose, because the uvula seal, the seal between this pressure gradient here and this pressure gradient here, it gets broken. Yes, no, maybe so? So the uvula keeps food from going into the upper respiratory or ventilatory, really, anatomy. Pretty darn important. All right. It looks like we're now going to be taking air from the nasal cavity back into the throat. So at this point, all of this air is going to get channeled and funneled through a single opening at the back of the nasal cavity. So if you draw this straight line right down past the sphenoid cavity, this is that sphenoid sinus. Maybe you guys remember frontal sinus, sphenoid sinus, I don't know. This is where mucus is produced and that drains down into your nasal cavity. If you draw this straight line down right behind where all the concha are converging, that is an opening. It's a passageway, but it's on the inside of the nasal cavity. So that whole area right there, it's not a physical structure, it's an opening, just like your nostrils are an opening. That's called a nair, and since it's on the inside, it's called an internal nair. That's also, by the way, known as the choana. Honestly, on a lab practical, feel free to use choana because sometimes it's easy because Moana. But it wasn't always like that. It's actually pronounced Kona, but let's see here. What do we got? We got same things. We got these different meatises. Here's that passageway, the opening down at the back of the nasal cavity. So that is our internal nair. Everybody feel somewhat comfortable with internal nair? It's a passageway. It's the open space. Inferior nasal meatus, we can label that at this point. Very cool. Okay. Now it passes into the throat, and the throat is called the pharynx. Okay, the throat's called the pharynx, but there's actually 3 different regions of the pharynx. And for this, I'm going to go to my PowerPoint. I just think it's easier. There's 3 different regions of the pharynx. The pharynx is this entire throat back here. The pharynx starts at the end of the nasal meatises, because that's where that internal nair is. It passes by the back of the mouth or oral cavity, and it's going to descend down to where the voice box is at which point it's going to kind of convert to the esophagus or the trachea, depending on which system it's going to pass down in. Air should pass down into the trachea, so it's going to have to pass through the larynx to get there. So that region of the throat, by the way, this is for both the respiratory and digestive system. Food is going to pass through this region of the throat as well. This is called the laryngopharynx. The area at the back of the throat, right behind the oral cavity, is called the oropharynx. And the region at the back of the nose, behind the nasal cavity, is called the nasopharynx. The nasopharynx is where we find the pharyngeal tonsil. This is actually a tonsil system that is part of our immune function. So as you breathe air, it should hit this tonsil way up here at the top of your nasopharynx. And that starts an immune function. It's going to start to break down bacteria and whatnot that gets into it. Oropharynx is just this region right here, just at the back of the throat right there. What we would normally think of as the throat is the oropharynx. People don't always think that the throat is going to go up and down from there. But we're not done with the nasopharynx. The nasopharynx also has the opening to the eustachian tube. You guys remember learning about the eustachian tube in A&P1, maybe? Right, the eustachian tube or the nasopharyngeal tube. This is important. It's right next to the pharyngeal tonsil. It goes into the nasopharynx. Did anybody, when you were a kid, or anybody, parents here with kids that were really prone to get ear infections? And that's because if you're breathing too many bacteria or whatever, right, that is going to build up right here in the pharyngeal tonsil. And if it ends up basically pharyngeal tonsil not able to fight all that bacteria can easily be transported up into the ear. So if you have a kid that gets like chronic ear infections and stuff, What's one of the number one treatments that doctors tend to do nowadays, and especially 20 years ago? A little bit less on the drug. Tubes, they're going to put in tubes, drainage tubes. Good, definitely. They're going to remove the tonsils. So does anybody remember the name of these tonsils? Other than pharyngeal tonsils, you may or may not have heard this, especially as a parent, because doctors tend to call them something different. Yeah, adenoids. Those are the adenoids right there. You got it. I love the context. Especially if you're a parent, you've heard the term adenoid before. And if you haven't, it's all right. The pharyngeal tonsil is also known as the adenoids, and they'll do an adenoidectomy. And they'll cut those tonsils out, not the tonsils at the back of your throat, contrary to popular belief. All right, so back to the throat here, nasopharynx. Nasopharynx is the region of the pharynx, the throat, behind that cavity. We good? So what's the function of the nasopharynx? Everything's got a function. One, it's just a passageway. Cool. We need a passageway to conduct air down into the lungs. But what if I told you that the nasopharynx was lined with a whole bunch of cells that secrete mucus? Again, we're talking about filtering particulate matter out, true? But here's something else that's going on inside both the nasal cavity and the nasopharynx. Is that what if I told you that air that makes it down into your lungs also needs to have 100% humidity? For proper diffusion of oxygen and CO2, the humidity of the air by the time it reaches your alveoli needs to be 100%. And so now as air passes by mucus, just think of water on your skin, right? And as air passes by that water actually enters the air. I don't know if you knew that or not, but it's what it helps cool you down. As the air takes the water away, the heat from the water, the heat from your body is going to dissipate out into the water. So you're starting to humidify that air. Yeah, swamp coolers, exactly. Does that make sense? As air passes through these tubes, both the nasal cavity and the nasopharynx and onward and upward, as it hits mucus, it's going to be humidifying that air. Yes, no, maybe so? So it's an important part of the nasopharynx. We're going to be talking about the epithelial tissue that goes along with that as well. In addition, now that it's entering your body, proper gas exchange needs to occur at body temperature. So a function of the nasal cavity and the nasopharynx, in addition to acting as a conduction system, filtering out immunes, dealing with the immune system, filtering out dust particles and bacteria and stuff, and humidifying the air, it also helps bring that air to body temperature. That's why anybody ever run in 0 degree weather? I'm not talking about 0 degrees Celsius, I'm talking about 0 degree Fahrenheit. Or, no? I used to have to. We couldn't row in the winters in Eastern Washington. Eastern Washington is not Western Washington. Eastern Washington is more like Montana. And so during the winters, we still had to practice. We still had to, build our endurance and everything. So we were out running about 10 miles every single day, no matter how cold it was. And often it dropped down to the zeros. It's really hard to breathe. It's super painful to breathe. And your chest feels just constricted all the time as you're trying to breathe this frigid cold air. And one of the reasons why is because as the air makes it down into the lungs, your body wasn't able to get it up to body temperature. And so the gas exchange is suboptimal. So you're having to breathe deeper and harder, and it really makes the chest, all the muscles of the chest are really getting overtaxed. Yeah. By the way, the same thing happens to people trying to run in extreme heat, because now the body wants to cool it down, and it really starts to take a shock on the system as it goes through. Crazy, crazy, crazy. On these models, there's a little dent. I don't know if you could even see it very well on this model, but there's this little impression right there. Can you see it right here? See how there's like this little swirl right there? That is the opening to the eustachian tube. Right there, that little swirl. That's the opening to the eustachian tube. You can see it on a lot of these different models, not this one. But that's the pharyngotympanic tube, the eustachian tube. I mean, people do that race every single year, and you know, it only kills a few people, so he should be fine. All right, let's do this. Let's do this together. Okay, I'm gonna just move all these things out of the way. Now you guys get to work together as a group. Here's what I want you guys to do. Everybody pay attention to the rules of the game. I'm gonna ask, what is this? You're gonna type it in chat, but you're not gonna press enter. Is that clear? The rules of the game. You're gonna type it in chat, but you're not gonna press enter. When I tell you to press enter, you'll press enter. You ready? And who cares if you're right? Who cares if you're wrong? Just play the game. What is this arrow pointing to? What specific structure is this arrow pointing to? Everybody type it in chat. I'm going to start my timers up here. All right, everybody go ahead and press enter. Perfect, that's the middle nasal meatus. Let's go ahead and get our label over on that so you guys can now take that off your list. What is this structure at the tip of this arrow right here? Everybody go ahead and type that in chat. What is this structure? Tip of the arrow. Again, pretend that you're taking a lab practical right now. So make sure you're not using abbreviations or anything. Make sure you're using the full term. All right, go ahead and press enter. Perfect, thank you. Nasal vestibule, fantastic job. That is where that hair grows from. All right, let's talk about this yellow dotted area right here. What is this yellow line indicating? Type it on in the chat. And please press enter. Good, I like that both Ashley and Melissa wrote pharyngotympanic tube. That right there, the yellow, is an indication of this area. It's actually looking at the tunnel, right? The passageway. Whereas the eustachian tube or pharyngotympanic tube, that's this swirl right there. I know they're so dang close to one another, but that area at the tip of my arrow, tip of the pointer right there, that's the pharyngotympanic tube. The one that I was after was the internal nair right there, okay? All right, just a few more things to do. Shouldn't be too crazy. There's actually not even a drag. Here, let me do this. I actually don't even think we need to go through the other two. I think you guys got this. There's going to be a superior nasal meatus, an inferior nasal meatus, and then of course we did internal nares here. So this open passageway here will be external nares. How'd you guys feel about that? You guys learned pretty quick, right? Cool. All right, so we've now gotten all of that air down through the tubes. That model's not great for it. Let's do in my, so this is better. So now we've gone down through the nasopharynx, the oropharynx, and the laryngopharynx, and none of these pictures did a very good job at showing oro versus laryngo. Bummer. Let's go to my pictures here because I'm guaranteeing that it will. Like this one right here. This is my Hemi head. I really like the Hemi head. The Hemi head now is going to be showing you more of that pharynx. So now, what would this area right here be called? If I'm looking at the throat, they normally referring to the oropharynx. That's right. It's incorrect, though, Heather, to be referring to the entire throat as the oropharynx. But yeah, that's definitely the case. Yep. So that area right there would be the oropharynx. And then, man, not even I did a good job at showing the laryngopharynx in a picture. Because why would I, right? Well, the laryngopharynx is right here. It's not too hard to see, maybe, but it's this region right, we'll flip that around, right there. So it's right at the lower portions of the throat, right at the entryway to the larynx. Make sense? So that's the laryngopharynx down there. Sweet. Awesome. Thanks, guys, for following along, playing the game, having fun. So from there, it goes into the larynx. The larynx is a really cool group of cartilage. This cluster of cartilage right here is going to help make up the voice box, but it's also going to help with the conduction of air down into these respiratory tubes. This larynx is suspended in place by a bunch of ligaments that you guys don't have to learn about. And those ligaments are attached to a really cool bone that's very unique. This bone is unique because it's the only bone in the body that is not perfect, Heather, that it does not articulate with another bone. This bone here does not connect with any other bone directly. It's hanging from its own ligaments. It's a floater. It's the hyoid bone. Now everybody can feel their own hyoid bone. Everybody take your hand, find your voice box, go just superior to that, and then you're going to pinch the lateral aspects, just gentle. Not trying to choke yourself. So you're going to pinch the lateral borders of your larynx, but go superior. So you're kind of right at the base of where your chin and your larynx are going to meet. Now do something you've probably never done before. Gently move that region of your neck back and forth laterally. You guys feel that kind of pop, really weird feeling? Or you just feel a pulse? Well, I can feel my hyoid bone very easily. I imagine once you guys start to get the palpation down, you will as well. You will be palpating people's throats. You'll have to be able to find areas around the hyoid, because just inferior to the hyoid is this chunk of cartilage right here. And just inferior to this chunk of cartilage right here is a major gland that you guys have already learned about. So take your own fingers, start moving it down, and you'll come into some fleshy, softer areas of your neck. What gland are we talking about right there? Don't worry, this is important, I promise. This will be helpful. Yeah, it's the thyroid gland, exactly. So this is the thyroid gland right here. And why that's important is because the thyroid gland is suspended from the inferior aspects of this big cartilaginous shield that's on the front of your larynx. This piece of cartilage, this is one huge piece of cartilage, is where the thyroid gland is suspended from. It's not only attached there, but that's where it starts. And that's why they call it the thyroid cartilage. Does that make sense? That big shield on the front, we're going to come back to some other pictures of this, don't worry. But this big shield piece of cartilage in most of your models is this really light blue. So anytime you see this really light blue, that's what we're dealing with. It's cartilage. This shield is the thyroid cartilage. Now, just inferior to that is what looks like a small piece of cartilage. And you're going to see that it's actually not as small as it looks at first glance. But right here at the tip of my pointer, if everybody please look at their screen, this piece of cartilage is covered by the thyroid gland and some muscles. And on this side of the model, they've been cut away. This piece of cartilage is independent. You can see there's like some ligaments above it and some ligaments below it. There's a little bit more space. This one right here is called the cricoid. Cricoid. Jiminy cricoid. Not a cricket. These are the two major pieces of cartilage that you can see from the anterior aspect of your larynx. Well, Heather, you guys don't need to learn the muscles around the larynx. Thank you and you're welcome. So do you really want me to go into it? Because it just is extra stuff for you guys to learn. Okay, good. Yeah. I mean, the names of all the muscles, most of the muscles of your body have to do with what they're attached to, where they're going to, whether it's going to an epiglottis or whether it's going to be going to the arytenoid, and where it's stemming from, which would be the thyroid. So something like this would be, this big muscle group that goes from the thyroid up near the epiglottis. So it's going to be something like thyroid epiglottal muscle. Pain in the patella to go through all those. Okay, anyways, thyroid cartilage and the cricoid cartilage is the anterior aspect of the larynx. So when we do a bracket like this on the drag and drop, what it's trying to get you to look at is the overall structure, right? So if I put a bracket around this on a lab practical and I said, what structure is this? I'm not asking for thyroid cartilage and cricoid cartilage. I'm asking for the larynx. Does that make sense? Let's continue on this bronchial tree, and then we're going to come back and talk about that larynx a little bit more. But air now passes down into the windpipe. Now, what did you guys learn about that windpipe? It's called what? What's the overall area? What is all of this called? Windpipe. Medical term for it. I don't know if I'm lagging or what. What's the medical term for the windpipe? The whole thing is the conducting zone. I love that you tried something there, Heather, but maybe I just didn't ask the question properly. But yeah, that whole area, goodness gracious, is the trachea. So what is this arrow pointing to? It's pointing to a single blue strip. And what did I say the light blue was in all the models that you've been looking at through the semesters? It's cartilage. Okay, there's these, and this is important, there are C-shaped, not circle. It's not a complete circle, although it looks like it on this particular image, on this view. But these are C-shaped pieces of cartilage. So they wrap around, but they don't connect in the back. Yes, no, maybe so? Do we know? What do we know? What do we not know?
Do we know why they're not connected in the back?
Oh, yeah. First show. I'll get there. So the first thing to think about with cartilage is cartilage, of course, is a harder structure. You're welcome, Melissa. It's a harder structure, right? Cartilage is firm. We see it in between the joints of our bodies, right? All the different bones are connected. Where they're connected, cartilage is found. Yes, no? So we know cartilage is firm. It's a structure. So what I want you guys to think about is air going through a paper straw. Why do we not see paper straws out there? Why is this world inundated in plastic straws? Because if you start to drink liquid, yeah, they suck, seriously. If you start drinking liquid through a paper straw, is there support? What's going to happen to that straw, y'all? Help me help you. What's going to happen to that straw if you're sucking through a paper straw? It starts to get wet, it gets soggy, but help me. One more thing, y'all. Come on. Soften for sure, but keep going. You take any tube and breathe through a tube that doesn't have structural support. What's going to happen to that tube? Breaks down? Sure, but I'm trying to get you guys to think about a tube that doesn't have structural support. So think of a plastic tube. Try to suck through it. It's going to collapse. Does that make sense? No suction, Parker, because what's going to happen is resistance is going to go to infinity. If you don't have structural support around a tube, and you're trying to create a pressure gradient, a suction of air through that tube will collapse. End of story. It's different for blood vessels. We don't need to worry about that for blood vessels because the pressure is coming from inside of the tube. We have a heart that's pushing blood through. That's liquid. Right now we're talking about air. In air, the pressure gradients that are going to be moving air through a tube are suction. They're vacuums, just like if you're trying to drink through a straw. Different than if you're trying to blow through that same straw. If you're blowing through that straw, you don't need to worry about it collapsing because you're forcing air down through it. But here we're going to be creating a vacuum. Yeah, so do mine. Obnoxiously so. Why did I buy you that drink? Flush money down the toilet if you become a parent. Might as well. Anywho, now, why is it C-shaped? Again, my lecture is going to be the best one for this. So as we start getting down into the trachea, here's what it looks like with a cross-sectional view. So again, light blue, what are we dealing with? I'm dealing with light blue. What is this? It's a cross-section or a cut across. That's the cartilage that we just looked at. Do you guys see how it looks like a horseshoe or like a C? Yes, no, maybe so? What's important about this? One, cartilage has structure, so it's going to keep the tube from collapsing. Two, inside of this trachea, on the back side of the C-shaped, we have a muscle. It's called the trachealis muscle.
You will have to identify this for your lab objectives. And it connects the ends of the cartilage. If we have a conducting system, can I change resistance of the tube if there's no muscle? Not very well, right? Because resistance is determined by the diameter of that tube. What's going to happen to the diameter of this tube if we contract this trachealis muscle? With the way that it's set up right now, we contract this trachealis muscle, then the tube will decrease in its diameter. Make sense, everybody? So we have a way to start to create resistance and change resistance just in that windpipe alone. It's not going to be as dramatic as, say, bronchial constriction and bronchial dilation, but it's a start. We got one big tube, and we can change airflow by messing around with this trachealis muscle. If the cartilage went all the way around, we would have no control over the diameter at all. Does that make sense to everybody? If the cartilage was circular, we'd have no control over the diameter at all. Yes, no, maybe so? One more bit that's kind of interesting and important, I suppose, when you really want to get down to it, here's a cross-sectional view of it in histo, is that the posterior border of the trachea is the esophagus. This is where food goes. And I want people to kind of think back at some of the times where they maybe swallowed something that was a little bit too big. And sometimes you feel like you're choking. But all you have to do is keep swallowing, and that food will continue to descend down through the esophagus into the stomach. It's not like the food gets trapped in the trachea. What's happening is that the esophagus is a big muscular tube, and it pushes against the space of that trachea, that tracheal cavity in there. Does that make sense? Yeah, trachea is super cool. There's so many cool things about it. For instance, down here is something called the carina. I've had lots of Karinas in my classes, like girl names, right? Karina, and I'm always like, hey, there's my Karina. Carina right here is at the very base of the trachea, and it's right where it branches into the bronchi. And the carina has a massive number of receptors, sensory receptors. Massive number of, just a huge cluster of sensory receptors that are going to detect mechanical changes. Primarily changes, any changes that they can find, but primarily irritants. So what about if we had a liquid irritant that dripped down there to the carina? What is your body going to do in response? Do we want liquid down in our lungs? Does that make sense to everybody? Right here at the carina, this is where our primary cough reflex is found. So if you start getting phlegm that's dripping down your trachea, rather than getting carried back up, which we'll talk about in just a second, when it makes it to that carina, it's going to cause you to cough. We're going to be getting into the histology of the trachea, but what you'll notice about the inside of the trachea is that it's lined with these big, long, hair-like projections. Those are cilia. Cilia are controlled by muscles, and cilia actually move. So all the cilia of your body move, like a sea anemone legs, they're moving. Okay, if I actually get you onto my fingers, they look like this, right? They're constantly moving. And they move in a direction. So they're going to be, say for instance, if something's on the surface of those cilia, and those cilia are going to be moving back and forth. They're trying to move whatever's on their surface in a very specific direction. So all the cilia of your body are like that. If you didn't learn that in AMP1, then I guess you're learning that now. And this trachea is lined with cilia. The entire trachea is lined with cilia. Because why? It's so cute. It's so cute. I had a student make this for me. It's so cute. I can't do stuff like this myself. What do you mean help? What happened? Oh, help move things. Are you guys with me on this? I'm getting a few people that are tuning out, I think. Right? So this is really important for moving things. The last thing that I'm going to teach you guys, and then we're just going to move on with the anatomy of this, is that this border here is called the mucosal layer. Mucosa. Mucosa sure sounds a lot like mucus, right? It's called the mucosal layer because these cells are designed to secrete mucus. Lots and lots of mucus inside of your trachea, which seems counterproductive to a lot of people at first. But the idea is to coat the border of your entire trachea with mucus. So now when things, particulate matters, bacteria, dust, anything that gets trapped in the mucus now, which is awesome, we want to trap it, now we take, and using the cilia of our trachea, we move it back up via an escalator service, mucociliary escalator, using the cilia to transport the mucus and then the debris back up. Cross section. X section means cross section. So cross section like this. You take a tube, like a straw, and you just cut it in half, and then you turn and look at it on the end. It's a cross section. Now, where does all of this mucus end up? Well, crap, it's got to go all the way back up through the larynx, all the way back up to the laryngopharynx, and then you swallow it. So all of that mucus from your larynx and from, well, I guess from your trachea and your larynx is going to end back up into the esophagus, down into our stomach, and then we digest that bacteria. Does that make sense? All right, let's finish this bronchial tree off. Where it branches, we call this bronchi, but the first bronchus and the second bronchus and the third bronchus we called primary. Versus secondary, right where it branches. So like, for instance, in this picture, this is a secondary bronchus, but so is, sorry, my bad. Trying to make this real quick for you. So is this region right here. Do you see how that would be a secondary bronchus? Secondary bronchus. Let's go over to this side. Secondary bronchus. Sorry, primary bronchus. And it branches to there. So right there is a secondary bronchus. True story? There's also a secondary bronchus right here. And it's really hard to see in this picture, but there's also a secondary bronchus right here. So go ahead and write this down. In the right lung, there are 3 secondary bronchi. But in the left lung, there are only 2 secondary bronchi. Yes, there's a reason. We'll come back to that. How many secondary bronchi in the right lung? We'll cover that again, don't worry. Here we go. 3 secondary bronchi in the right lung. How many in the left lung? 2. Sweet. Tertiary bronchi are the colored portions here. So if I show you this model, hint, hint, and I point an arrow, hint, hint, at the colored ends, hint, hint, those are tertiary. And you can see there's lots of different tertiary, okay? Lots of different colors. Those are all tertiary bronchi. Yes, no, maybe so? Double it up, double the fun. What's different? Oh, primary versus lobar versus segmental. Okay, let's just do those. Left primary bronchus, we kind of already went through that. Each primary bronchus goes to a lung. There are 2 primary bronchi. Why? Because there are 2 lungs. There's a right lung and a left lung. Make sense for primary bronchi, yes? How many did we just walk through? How many secondary bronchi are there on the right? 3. And how many on the left lung? And that's because they're also known as lobar bronchi. And if you come down and look at the lung, what you'll notice is that the right lung has 3 lobes. It's got a lobe up here, a lobe here, and then there's also a lobe down here. Yes, no, maybe so? 1, 2, 3 lobes. How many lobes do you guys see on the left lung? And that's why there are 2 secondary bronchi to the left lung and 3 secondary bronchi to the right lung, is because each secondary bronchus leads to a lobe. So they're also known as lobar bronchi. Does that make sense? Secondary and lobar are the same thing. It just has to do with what lobes they're going to. Now, from the lobes, each lobe, and you don't get to see it in this picture right here. Nope, no, Trisha, no, it's just, we just changed the size of the passageways. Great question. But you'll notice that the passageways are not the same. You see how the bronchi are not the same size, not the same length. Okay, we're just going to change some of the anatomy to change the flow. We want the flow between the left and right side to be equal. Great question. So in order to understand that point a little bit better, right, because we go from this bronchial tree, primary, secondary, tertiary, quaternary, we go down into the lungs themselves. What you'll start to notice, I color coordinated some of this stuff, is that each one of these segmental bronchi, so that's going to be your tertiary bronchi. What's going on with the segmental? I might have taken this out of this lecture. Oh, that's funny. Why would I do that? Ah, no, it's down here. Whew. Okay, so segments. You do not need to know all of these segments. Did I make myself clear? You do not need to know these segments. End of story. Absolutely do not need to know all these segments. I hope I made myself clear on that. But each one of those segmental bronchi go to one of these different colors. These are called segments. So inside of the lobes, for instance, there are 2 lobes on the left lung, but there are 8 to 10 segments, just depends on the person. So in the superior lobe, you can see that there is a segment here, a segment here, a segment here, and a segment here. Yes, no, maybe so. You don't need to know their names. Just understand that there are different segments. So what is a segment? Yeah, you can have different number of segments. What exactly is a segment? part of a lobe, but it's more specific than that. Each segment is separate from the other segment. It's a beautiful evolutionary adaptation of the lung. Each segment is separated from the next segment. Each one gets its own segmental bronchus. Each one gets its own pulmonary arteries and veins. Each one is surrounded by its own membrane. It's completely separate from the other ones. Why is that important? Because if you get a bacterial infection in this segment, do you want that bacterial infection to easily spread to the rest of the segments? Exactly, it's self-surviving unit. It's really cool. So now you got somebody with lung cancer, and that lung cancer is isolated, for instance, just to this anterior bronchiopulmonary segment. What can we do? We can go in and we can excise just that segment, leaving the rest of the segments intact. And therefore, lung function won't be cystectomy. You've been in this field, so you know what the heck I'm talking about. Right? Does that make sense to everybody? That we can act, it's a beautiful thing that we can kind of take advantage of, especially with lung cancer patients or lung failure patients. We can go in and try to remove sources of problems while helping to maintain the rest of the lung. It's really cool. So you guys don't need to know about all the different segments. You don't need to know all the names of the different segments, but you need to understand the basic function of all of those segments. Is that clear? And that's why these are called segmental bronchi, because each one of those bronchus is going off to a different segment. Make sense? So the tertiary is also known as segmental. Lovely. We can fill the rest of this in. We already know this. The bone up at the top is hyoid bone. The cricoid cartilage is on the lower area of the larynx. The superior cartilage is called the thyroid cartilage. Starting from the thyroid cartilage and descending all the way down to cover the tracheal cartilage is the thyroid gland. And of course, that big long windpipe is the trachea. How do you guys feel so far? We're almost done, honestly. It's not that bad. And we're going to use our flow chart to discover how we can study this better. Let's do one more model, and then we'll take a break. We're just going to go ahead and kind of continue this idea with the lobes, because this looks like it has a lot going on, but this slide is actually really darn easy, in my opinion. And I think you guys will end up agreeing. So for instance, what is this blue cartilage rings? What is this tube right here at the tip of the arrow? Go ahead and just write it in chat. We don't need to wait. What is this? I think I'm having a little bit of a lag, but that's okay. There are two regions of your lungs. There's an apex and there's a base. Now, this can be a little tricky because students learn that the apex of the heart is inferior and the base of the heart was superior. But this one makes a little bit more sense in my mind because apex tends to be where the peak is found. And if you look at this right here, there's a peak of the lung at the top. So this is the apex of the lung. Whereas down below, right where that lung meets the diaphragm, this big, huge muscle, one of the biggest muscles in the human body, that area, that region where that lung meets the diaphragm is called the base of the lung. Now, in terms of the lobes, they're really not that hard. If there are 3 lobes, then I have a superior, an inferior, and something in between. Whereas if there's only 2 lobes, then I have a superior and an inferior. There's nothing in between. So the left side has a superior lobe and an inferior lobe. But since there's nothing in between, there's no middle lobe. Does that make sense? Whereas the right side has 3 lobes. It's got an inferior, it's got a superior, and then it's got something in between. So we've already used most of our arrows. It's been pretty darn easy. Separating each lobe is a big crack. And just like in the brain and the spinal cord, these big cracks throughout the body are called fissures. Oblique means angled, in case you didn't know that, right? An oblique means an angle. So like your oblique muscles were angled muscles. So when you're looking for an oblique fissure, you're looking for a fissure that is angled. Does that make sense? And in your left lung, you only have one fissure, and that fissure is very, very angled. It's got almost, this 45-degree angle, really, if you draw a line. Now, I want to make sure you guys understand that the oblique fissure is not only found on the left side. This model here doesn't, the drag and drop here doesn't do a great job at showing that. But do you guys see that there's also a fissure down there, right at the tip of that tiny little blue line? It's so little. All right, now I got a bigger blue line. But at the tip of that blue line is also a fissure, and it's also at an angle. So on the right side of the heart, it also has an oblique fissure. Yes, no, maybe so? And we can just put that in so when you guys take a screenshot. Just don't want you guys to lose sight of that. Now, there is also a fissure that runs pretty much flat. Sure, it angles here at the end, but there's a big chunk of this fissure that is on a horizon, right? It's going to be a horizontal fissure. Each lobe is made up of lots of little lobules. So each one of these little, irregular circles, those are called lung lobules. Lobules are where a lot of the action happens. So this would be kind of a representation of one lobule, multiple alveolar sacs. So this is where all that respiration is going to happen. The last thing has to do with why the right lung looks different than the left lung. And it has to do with the orientation and location of the heart. You may or may not remember this from the narrated PowerPoint, but your heart does not sit in the middle of your chest. It starts in the middle of your thoracic cavity, but then it lays where the apex is pointed left and anterior. So the apex of the heart is right here. And it ends up taking up a big chunk of space where that left lung would be. Does that make sense? So therefore, right at this location, we need to make room for the heart. And so the lung, it looks smaller, but its function, it ends up kind of filling a whole bunch of other different types of space. But right at this point where the heart fits into that lung, we're going to need to make space in that lung for that. And that area right there is called the cardiac notch. So that border right there is the cardiac notch. If I was talking about the inside of that, this is technically the fissure here, but if I start to look at the actual shape right there, Right there, that physical shape, that's the cardiac notch. Whatever. I'd probably end up testing you on something like this, and you can just call it the cardiac notch. Same thing, superiors and inferiors. I'll let you guys label this one. It's the exact same thing, but what we've done is we've cut away part of the lung. Easy peasy. This one right here starts to talk about some of the other structures that students have trouble with. Right where the trachea, bronchi, primary, secondary, blood vessels, nerves, and nerves, right where they enter the lungs and leave the lungs. This is a spot called the hilum or hilum. This technically would be the hilum of the lungs. And this one here would be of the left lung. And then there's going to be a hilum of the right lung over on the other side. What you're going to find is that something like your kidney also has a hilum. Here's my funny way of remembering it is that anytime you look at these organs right here, you'll have this kind of area of the organ where things are, being able to go in and out of this little space right here. But when you look at the organ, a lot of times these organs, especially the kidneys, right now, if you look at this kidney right here, goodness gracious. You look at this kidney right here, you can see that there's an area where you got this big blood, this tube, and the blood vessels going in and out, and nerves going in and out. This would also be considered a hilum, yeah? Right there, that space right there where all of these things are going in and out. Does that make sense in terms of why, oh goodness, go back here, why this would be considered a hilum? They're the same idea. My funny way of remembering it is it kind of looks like a butt crack. So where the butt crack is at, it's the space. That right there is the hilum or hilum of the lung and the kidneys. So do what you got to do to remember things sometimes. Let's take a break. It's 12:15. We're almost done. It's just I'm hungry and I need to go to the bathroom. We still need to go through the larynx a little bit more. Then we're going to finally make it down into one lobule. We also need to make sure that we understand how the lung, and we're going to go through the entire physics of what's going on here tomorrow, but we need to be able to talk about the anatomy of the pleura and and then some histology. Ugh. Okay, let's take a break. We won't make it too long. Go grab a bite to eat, bio break, something to drink, and be back at 25 after. All right, let's finish this off. Let's finish this off. We got this. Power through. I am going to just quickly show you guys this really cool little picture. I like this one quite a bit. Is that basically we've had a bronchi, mini bronchi that are branching and branching and branching, but we're just following one part of one bronchus down here. And you'll notice that the cartilage is starting to thin out. We don't got these big, massive bands of cartilage anymore as we get down into the smaller bronchi. And finally, these smaller bronchi are just going to keep on branching and branching and branching, and the amount of cartilage is just decreasing this entire time. We're now in such small tubes that the pressures inside the tubes aren't going to be as big because we branched out so many times. We're dispersing these pressures through lots of surface area. So at some point, basically the cartilage, it's not going to disappear. This model's a little, or this picture is a little bit misconceptive here, but it's not just going to disappear, but the amount of cartilage is really darn small. And by the time we make it to the bronchial and the terminal bronchial, you can basically just say, for all intents and purpose of things, that cartilage is gone. Can you do me a favor and write that down somewhere big, bold, that you can remember? This will be important, especially when we get to the histology. Is that bronchi, definitely cartilage. Relatively disperse. You get to the bigger bronchi, we're going to have tons of cartilage. Smaller bronchi, cartilage. But one of the things that differentiates bronchioles from bronchi is basically the lack of cartilage. It's also going to have smooth muscle that surrounds it, so we can start to really impact the diameter of these tubes. These are going to be covered in adrenergic and muscarinic receptors. Not just adrenergic. These are not blood vessels. These are bronchioles. So the types of receptors that are found here are going to differ. We'll get more into that in tomorrow's lecture. The bronchiole branches becomes terminal bronchiole. This is the end of the road for the conducting pathway, and that terminal bronchiole is going to branch, and each branch is going to go off to this little alveolar cluster. Okay? Each portion of this cluster is a different alveolar sac. So in this particular picture, there are 1, 2, 3 alveolar sacs. And in order to get there, it's going to have to go through a respiratory bronchiole, which then becomes an alveolar duct, which then leads to each individual circle, which is called an alveolus. You guys might remember this from our flow chart. Terminal bronchiole, respiratory bronchiole, alveolar duct to the alveoli. Yes, no, maybe so? Cool. Let's finish this up. Let's finish this up. What's going to end up happening down here is that you get this really beautiful structure, this really cool, basically, branching of tubes. And you'll notice that this terminal bronchiole in this picture, it's really well done. Do you guys see all these circular bands of muscle around that terminal bronchiole? Lovely, 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 because structure determines function. If I don't have a lot of cartilage, but I do have a lot of smooth muscle, now I can really start to mess with the diameter of these tubes. What's going to happen to the amount of smooth muscle as I go down into the respiratory bronchioles? It's going to start to decrease. We're going to be dealing a little bit less with the dilation, bronchial dilation and bronchial constriction down here. It still can occur, but it's not as functionally important. Now we get to the alveolar duct and we lose all of that smooth muscle. It's just simple squamous epithelial tissue, super thin, and diffusion, respiration really starts up down here. Alveoli, that is their function. It's gas exchange. So there's just a massive amount of anatomy related to the physiology. Each alveolus is surrounded by its own capillary bed. every tiny little circle, and these things are super small. You have multiple millions of them in each one of your lungs. And each one gets its own capillary bed. Imagine this basically having a ball that's surrounded by string, right? You're just wrapping each alveolus up with string. Now, in addition to the string, by a hairnet, I like that. In addition to that, we also have all of these elastic fibers. Every single alveolus is surrounded with rubber bands. Elastic fibers. Yes, elastic can stretch, but that's not what it means, right? Elasticity means the ability to what? Think of blood pressure. What does elasticity mean? Thank you, Beck. That's exactly right. It's the ability to recoil. Yes, no, everybody? So if I fill each one of these alveoli up with air, and then I release that air, the alveolus should return to its original shape. Does that make sense? What this means, and this is going to get even more significant tomorrow, is that the alveoli of your body, every single alveolus is trying to recoil right now. Let's put this in a more extreme way of thinking. Every single alveolus inside of your body right now wants to collapse. It's trying to collapse. Your lungs are constantly trying to collapse. You're welcome. It's pretty creepy. Your lungs are trying to kill you right now. They're also trying to keep you alive. Don't worry. This is going to be exponentially important when we start talking about pressure gradients. It's going to help create pressure gradients that create vacuums. We must take into context these elastic fibers. Yeah, they're interspersed. They're interspersed. They're not independent, of course. This is just a model. The interspersed means that basically, you know, sometimes the elastic fibers go over the capillaries, sometimes they're under the capillaries. It's just, you know, it's a maze. Cool. Big old veins leaving. I lied. Big old veins coming off, oxygenated blood returning to the heart. See, even I get this messed up sometimes. That's why we want to make sure to think about the function of the lungs, this gas exchange. Big old arteries, they're blue, bringing lots of poorly oxygenated blood into the lungs, and we oxygenate it and get it the heck out of there. Sweet. Let's keep zooming in. Alveoli, the alveolar ducts are just simple squamous tubes, and they're going to lead down into each individual alveolus. It feeds the air through these tiny little holes called alveolar pores. Alveolar pores branch off from the alveolar ducts. We'll come back to the histology. But yeah, there's 300 to 400 million alveoli in each lung. Each one gets its own capillary, and each lung is about the size of a tennis court. So that means that your two lungs combined are about 2 tennis courts in terms of their surface area. It's absolutely insane to think about. Take all these tiny little circles and you roll them out. The surface area is ginormous. Each alveolus is surrounded by elastic fibers and capillaries. Huge part of its function. Smooth muscle, be thinking about how structure determines function as we go through this. And last but not least, we will deal more with this later, is that an alveolus, you're not done learning the anatomy just because you learned what an alveolus is. Each alveolus has its own world too. There are different types of cells that make up your alveoli. Type 1 alveolar cells, type 2 alveolar cells, and macrophages. Macrophages are a part of your immune system. Type 1, there are 99% of all the cells of your alveoli are type 1. So what's the major function of the alveolus? Go ahead and write this down. Major function of alveolus is gas exchange. "99% of the alveoli are comprised of type 1 alveolar cells. So if the primary function of the alveoli is gas exchange, what do you guys think the function of type 1 alveolar cells is? Gas exchange. So type 1 alveolar cells are just simple squamous epithelial cells, and they're going to allow for rapid gas exchange. That's their function. Pretty easy function. Type 2 alveolar cells are where things start getting weird. Type 2 alveolar cells secrete a chemical that we're going to learn more about later called surfactant. I'm going to write this in chat just so you guys have it. Type 2 alveolar cells secrete surfactant. You can start looking it up if you want. It's pretty cool. It's basically like a phospholipid. It's soap. Your lungs produce a soap, and we're going to learn how that is constantly helping your lungs from collapsing, because the elastic fibers tell your lungs to collapse, so we better have something that is competing against that collapse. And surfactant is going to be one of those things. All right, so let's go back down to our models here. And using the information we just learned, let's fill out our lab objectives. Shouldn't be too crazy. Shouldn't be too crazy. So leading down into this lobule, right? Leading down into all this, these are going to be bronchioles. Bronchioles, we're losing all of that cartilage. This model is a little bit, well, I think it shows too much cartilage, personally. But that's my opinion. There should be no cartilage down here, but on this model, there is a little bit of cartilage. It's a little bit confusing. But this bronchiole right here is going to branch. And it's going to terminate this system, right? So this right here is a terminal bronchiole. Terminal bronchioles are going to go out to different lobules. Yes, no, maybe so? That's going to lead down into the respiratory bronchioles. So where's the alveolar duct? Much more difficult to see here. Basically, it's inside of all of this mess. So what I'm going to point out is this black circle right here. That black circle, let me change this outline a little bit. Let's change its color so it becomes something unique. That black circle right there is the alveolar pore. And the alveolar pore stems off it's actually part of the alveolar duct. Let's just go there. Let's just go straight there. This is the alveolar duct. It's just one tiny little view of the alveolar duct, but that alveolar duct is inside of all this mess, and it's going to be moving through each one of these alveolar sacs, and it's going to be feeding each one of the alveoli through tiny little microscopic pores. So if I ask you guys on a test question, what is that black dot? What I'm looking for is alveolar duct. Is that clear? I spelt it wrong. Alveolar duct. There we go. Cool. Awesome. Wonderful. Now, those alveolar ducts are feeding down into each individual circle. It doesn't matter which circle I point at. Alveolus, alveolus, alveolus. Each one of those are alveoli. We start to add them all together, right? Plural is alveoli. This cluster of, grapes right here, That's an alveolar sac. Each alveolar sac is going to be fed by a different respiratory bronchiole. The capillary beds, pulmonary capillaries, technically, they're going to be surrounding each one of those alveoli. Red and blue. Don't forget that red and blue in the lungs are opposite of everything else. Poorly oxygenated versus oxygenated. Poorly oxygenated blood is leaving the right side of the heart to go to the lungs. So if it's leaving the heart, it's called a artery. Everybody feel pretty comfortable with that idea? And if it's going back to the heart with oxygenated blood from the lungs, it's a vein. The last thing to show is on this model, the surface of this model out here is a depiction of the outside of the lungs. We're on a very, very outside edge of the lungs. In the outside layer of the lung, the very, very outside, which is this light white line right here, that's actually made up of pleura. That's a membrane. And the pleura that comes into contact with the organ is called the visceral pleura. We covered this earlier this semester when we were talking about the heart, where we reviewed parietal versus visceral. Yes, no? I don't want to waste a lot of time here today. But viscera meaning organ. So the pleural membrane that comes into contact with the organ itself is called visceral. In terms of your lungs, they're not cardial tissue anymore, right? So we're not going to call it visceral pericardium. It's lung tissue, and lung tissue is all about pleura, pleura, and more pleura. So, yes, Heather, arteries and veins are opposite color, or not opposite, but they're opposite of what you think because we're dealing with lung function, leaving the right side of the heart to go out to the lungs. Arteries and veins are not defined by whether they're carrying oxygenated or poorly oxygenated. They're defined by whether they're leaving the heart or returning to the heart. All right, so if I have an arrow pointing to this outside structure anywhere, even if it's out here, what am I looking for? I'm looking for visceral pleura. Is that clear? So that leads me to this picture right here. So this whole thing is the thorax, thoracic cavity containing the lungs and the heart. And the heart's in that mediastinum place. And your lungs are actually adhered to the body wall of the thorax. Okay, you got your ribs and your different muscles. And even the muscles in between the ribs, which we'll be dealing with, intercostal muscles. But there are membranes that help keep the lungs inflated. Man, we are going to go into so much detail about the physiology of this inflation process tomorrow. So you guys better understand. You better understand the anatomy of these pleura. So just start there. If I have a membrane that's attached to the body wall, physically attached to the body wall, not attached to the organ, then this is going to be parietal. Yes, no, maybe so? If it's attached to the organ, which is what the white line is there depicting, it's attached to the lung, then it's going to be visceral. And the space in between, just like we learned with the heart, is a cavity. Pleura are unique because pleura secrete fluid. They secrete well, I shouldn't say pleura. Serous membranes are unique because they secrete serous fluid. Pericardium, visceral pericardium, and the parietal pericardium, they secreted serous fluid, and it filled up that pericardial cavity. Well, here we have pleural cavity filled with serous fluid. We'll talk more about the physiology of that tomorrow, but I believe earlier this semester, didn't I do the experiment, right? Two pieces of paper without fluid. I showed you guys this under my DocuCam. Yes, I think, right? I did. Help me. Am I here? Are you there? Are we there? Do you guys just not remember? Okay, there's some yeses, some major lag going on. You're not there, Heather? Is that what you're saying? You don't remember? Okay. No worries. I'm fairly sure I did, but if I didn't, I'll do it tomorrow. Not a problem. It's a super simple. Actually, you guys can do it right now when you get done here. Take two pieces of paper. Paper towel is a great way to go about this. It's better than just regular paper. Just take two pieces of paper with no moisture whatsoever. Are those two going to stick together? Are those two pieces of paper towels going to stick together? Yes or no? No, 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 no. So now you take a little bit of moisture and you put it between those towels. Just put it on one of those paper towels. Is that other piece of paper towel going to stick to it now? It's called surface tension. And so there is a surface tension between the visceral pleura and the parietal pleura. And don't you think that's going to help keep these two layers adhered to one another? I hope you guys are seeing that the answer is yes. There's a lot more to it than this, but your lungs are highly elastic. So your lungs are always trying to collapse. Your lungs are always trying to collapse. Your lungs are trying to kill yourself, to kill you right now. They're trying to collapse. So your body better have some things in place to keep those lungs inflated. I hope that makes sense. Well, we're running out of time. Let's get back up to this larynx here. So the larynx can be a little bit confusing. So this right here is the very, very top. It's the larynx. You can see on this model that I can take and I can cut it in half. So that's exactly what this next picture is. It's the very top of the larynx where I've taken a portion of that model and I've removed it. And then I just look at the inside of each one of those. Does that make sense in terms of what we're looking at here? Down below is going to be the trachea. The cartilaginous rings that surround that are tracheal cartilage. So we put this tracheal cartilage right there. Well, it turns out it's also pointing at that cartilage right there. Cool. We're going to be going more into the cartilage. I'm going to show you guys on a couple of other models and it'll make a little bit more sense with the cross section. But what I want to focus on here for this particular picture is that there's these funny little, I don't know, pieces of funky tissue, for lack of a better word, that kind of just look like weird folds of membrane. You guys see that? These dark gray lines are pointing to them. So what's going on there? What's going on there is that these are your vocal cords. So when you look down into the larynx of a patient, you're going to see something that looks a little bit like this using a laryngoscope, right? Laryngoscopies. We don't want to do those too often. They're kind of weird. Feeling tubes go down into your throat's never very fun. And there's a space between these membranes. That space between the membranes is called the, well, rim glottitis for, that's what's on here. But you guys can just call it the glottis. Glottis. Glottis is a space. It's a physical space that I can change the size of. You can see it in this picture right here, that the glottis on the right, it's going to involve both the vocal fold, which we'll come back to in just a second, and the rima God, I can never say this word, glottitis. But that space is the glottis. So just like your pupil is a space, and we can change the size of the pupil in your eye, the glottis is a space, and we can change the size of that glottis. Does that make sense? We change the size of this glottis based on the muscle groups that are actually attached to portions of cartilage that can move back and forth. It's super crazy. But right on the ends of some of these cartilages, you're going to learn what these cartilaginous names are in just a second. But you can see that right on the end of this crazy, funky, almost like a seal-like cartilage, you know, two seals, sea lions kissing one another, there are ligaments that extend all the way out to the cartilage up in front. These are called your vocal ligaments or your vocal cords. So this white line right here and this white line right here, these are depictions of your vocal cords. So when I look at this from the side, when I've cut this out, what you'll notice is that there's a white line that crosses right here. And there's a white line on the other side that crosses right there. These are your vocal cords, otherwise known as your true vocal fold. Now, right above, or I guess lateral to this, a little bit superior and lateral, do you guys see how that here in a real patient, there's something else that kind of looks like a folding of the skin right there? And when you look on this depiction over here, that's what this big flap of tissue is right here. On either side, these are called false vocal cords, but they're better known as vestibular folds or vestibular cords. Okay, these are just a roof. It's just another type of vestibule, and they help hold everything together. So these vestibular folds. So when I go back to my model, this flap of tissue on the top, this flap of tissue on the top, that's your vestibular fold. And I definitely want to go through that with y'all because students get these wrong all the time. They look at this model in the lab practical and they're like, they just invent words. Or literally sometimes just write, I don't know, Dr. H. What am I looking at? So I hope I helped you guys this semester be able to identify what this model is even looking at and how we have lots of cool lecture slides to help us understand this a little bit better, okay? Now, the next few models are models of the larynx. Now, we need to be able to look at this larynx in multiple different angles. We have an anterior view, I have a lateral view, and I have a posterior view. Each one of these views, you're going to be able to see the cartilage change its shape. So for instance, if we start on the front, the anterior view, and I'm looking at this big piece of cartilage that the thyroid gland is attached to from the top here, you know, the very inferior aspect of this piece of cartilage, that's going to be the thyroid cartilage, as I described earlier, this big, huge shield of cartilage. Yes, no? And it looks like a ginormous piece of cartilage. And I mean, it's pretty big. Don't get me wrong. But as I turn it laterally, this is the anterior portion over here. So as I turn this thing laterally, there, I can tell it's the anterior portion. By the way, if you guys want to know the landmark, it's the hyoid bone. You see how there's Dracula fangs on the hyoid bone? Good old Dracula. Those Dracula fangs are on the anterior portion of that hyoid bone. So as I turn it, there's those Dracula fangs. So I know that this side is anterior and this side is posterior. I'm a big fan of landmarks. Thyroid cartilage, thyroid gland, tracheal cartilage. You guys have already done that a few times. Okay, cool. Now let's turn it and look at the posterior view. The posterior view, you can see there's a lot more happening. It's opening in the back. It's kind of interesting, huh? But what is lacking? What did we just lose right here? What do you not get to see a really good view of anymore? What piece of cartilage disappeared, went bye-bye? Good. So the thyroid cartilage is a gigantic shield. It's closed in the front, but it's open in the back. Does that make sense? All right, cool. So let's go back to our anterior view. Now, between the thyroid cartilage and the tracheal cartilage, there was something that I described earlier that, you know, Jiminy Crickets, Jiminy Cricoid. There's something in between right here that's the cricoid cartilage. Cricoid. Now let's look at what happens to the cricoid as I start to rotate this. Of course, this is a picture that doesn't show it. I didn't. Well, it is what it is. Let me show you guys via this. Now, if I start to rotate this picture right here, this is anterior, this is posterior, but this is kind of this oblique. This is like slightly from the lateral view. The cricoid is way down here. If I look at this from a posterior view, look what just happened to the cricoid cartilage. It became much, much bigger, didn't it? It continues to circle all the way around. The anterior portion of the cricoid is very small, but the posterior portion of the cricoid is really, really big. Yes, no, maybe so? Can you guys see that in this picture? Cool. Can't see it in that view. But if I turn this around, what is this big piece of cartilage right here that I'm outlining? What is this big piece of cartilage going to be? This can be really tricky for students. I hope I'm helping you and not just boring you guys. I'm trying to help. I'm trying to help. Right? Cricoid looks really small from the front, but if I look at this same model from the back, this is the cricoid. Tracheal cartilage. The side of it is tracheal cartilage, but not the back of it. All right, so now we got to learn just the last couple pieces of cartilage, and then we'll be done with all this cartilage. What was the name of the space, the hole, that we can change the size of? What was the name of this space? No, not epiglottis. What's the name of the space? Ashley got it. Yes, Heather, I just saw your question. Yes, that's the only cartilage that surrounds the whole larynx. The space is called the glottis. Epi means above. Epi means above. So there is a flap of cartilage that lies above the glottis. Okay, so that's going to be called the epiglottis. You can kind of see it in this picture right there. It's this big thing right here. Glottis is going to be the space. Sometimes I like to go all the way back to a picture like this, because I think this is a really good picture for showing you oral cavity in the tongue. And then look right there. Fill this in with some red and some big weight. So what you'll notice is that the epiglottis, the epiglottis right here, that's that light blue. And it's a flap of cartilage that lies over the top of the glottis. So everybody swallow. What just happened to your tongue? Every single time you swallow, your tongue depresses and it pushes posteriorly. Yes, no, maybe so? And what's that going to do to the epiglottis? Well, it's going to cause this epiglottis to fold down. Every time you swallow and you move that tongue, it's going to move that epiglottis down like a door. It's like a really cool cartilaginous valve. Very powerful, very strong, and it's going to cover the glottis. So every single time you swallow, the glottis should be closed. Sounds pretty important, because do you want food and liquid traveling down into your respiratory system? Hell no. So what major structure blocks food and water from going down into the respiratory system? Everybody, what major structure blocks food and water from going down into the respiratory system? Sure sounds like something that you're going to get tested on, right? So when I look at this from the anterior view, the epiglottis sticks up and above, but the epiglottis is this big, I don't know, tongue-shaped piece of cartilage, and it's movable. It's attached to a bunch of ligaments, and it's very, very movable. And it covers this glottis as we swallow. Sweet. So if I come back here, I can see this epiglottis from a number of different views. It's hidden in this particular view, and then in this view, I see the epiglottis up here. Cool beans? All right, the last couple cartilages are the trickiest. There are a few pieces of cartilage that you guys do not need to know. For instance, the cuneiform. I have no model that can show you the cuneiform cartilage, so you will not have that piece of cartilage on your lab practical. However, the corniculate and the arytenoid. Students have a hard time pronouncing this one, so everybody say arytenoid. Arytenoid. Super boring, dry. Arytenoid. Imagine like Bueller. Arytenoid. That's the only way I can remember it, is by just saying it over and over. I got nothing else that goes along with it. But the corniculate and the arytenoid, yeah, not arachnoid. Arachnoid is obviously more, spider-like, but I like that you're making a connection. Any connection that helps. So the way this all works is the arytenoid and the corniculate cartilages, they are on the posterior border of the cricoid cartilage. The cricoid cartilage wraps all the way around. On the anterior portion, there's nothing attached to the cricoid, but on the posterior portion, the arytenoid and the corniculate basically are back here on the posterior aspect of the cricoid. Now, only the arytenoid is attached. So let's look at it from this view here. Still not great. Let's look at it from this view here. Still not great. Sometimes, I swear, these models are supposed to be to help you. Ooh, let's do my pictures. Little good. Little good. This is the cricoid. I still can't really, oh, there it is. I don't know if you guys can see this. Do you guys see how there is a line right there? It's not very easily seen. Aha. So that line is the border between two pieces of cartilage. So this piece of cartilage is attached to this piece of cartilage. Nice. Okay, it's not great, but at least it gives you guys a view. Sorry, it's not better. I blame my personal assistant. I know, you got to zoom in, and I told you it was there because I'm, the boss, and yeah, I get it. I get it. But what we're looking at here is this. Attached to the cricoid is the arytenoid. Attached to the arytenoid is the corniculate. And now attached to these are muscles and ligaments. And these muscles are crossing muscles. So if I contract these muscles, these oblique arytenoid muscles, what it's going to do is it's going to pull those arytenoid cartilages closer together. So imagine taking this right here, this arytenoid cartilage, and I'm going to attach one muscle group down this way. That's not going to be the best way to depict this. I hope you guys, I wish I had like an ability to draw on the screen. But if I'm attached like this, if I contract this muscle, it's going to pull this arytenoid this way. Yeah, I could. But then I don't know how to get that so that I can write on it. I mean, I guess I'd print it out and all that stuff, but I wouldn't be very fast here in class. Wish I just had like a drawing pad, but those are crazy expensive. And ACC won't buy me any of that stuff, and I don't have enough money. First world problems. All right. So it's attached here, and if I contracted this muscle, it's going to move the arytenoid in this direction. Yes, no, maybe so? Cool. Now I take this exact same idea, but I attach a muscle this direction, and now I got these crossing muscle groups. As each one contracts, it's going to pull these arytenoids together. And what that does is it's going to change the tension on those vocal ligaments. If I change the very tips of my arytenoids, then I change the tension on those vocal ligaments. True story? Does that make sense? Are you guys kind of seeing that I can make these vocal ligaments tighter or more loose? I can open the glottis or I can close the glottis. I got muscles that do that. You guys do not need to know these muscle names. Lots of different muscles help with the opening and closing of the glottis and therefore tension on that vocal cord. But the vocal cord is just like a reed instrument. Not everybody's played a reed instrument before, but like a clarinet or a saxophone. The way those work is that you blow through that reed. And it's the tension, it's like the air flowing against the tension of that reed that creates a noise. And a baritone saxophone versus a clarinet, what you have is different tensions on different reeds, and it creates different pitches depending on what instrument you're playing. Does that kind of make sense? I know not everybody in here is probably a musician, but I used to give this example of blowing in grass. I grew up where, I don't know why, my dad taught me really young that you can pick up grass, you can change the tension of grass between your two thumbs. Like this, you just put the grass right here in between. Yes, this is my kid's Band-Aid, deal with it, cut myself. And then you can actually change the tension on that grass by changing how much tension you put onto your thumbs. And you can change the notes that are coming out of that piece of grass between your fingers. Well, that's what your vocal cord is. Your vocal cords change their pitch depending on the tension that's being placed on them. So singers, anybody in here a singer? Like taking singing lessons or whatever? Anybody here like music? Anybody here? Hey, anybody? Yeah, Tariq, I was gonna say, anybody in here is like the bathroom or the car singer? Like you're driving down the road and it obviously you have perfect pitch. Like there's no better acoustics than the shower. No better acoustics. Car, same way, man. People are looking at you while you're singing. You look over at them and you just keep singing like you're giving them a melody. Like, who cares? You're never gonna see them again. Just keep singing. But if you ever took vocal lessons, they tell you that your voice is a muscle and you got to exercise it. If you're going to be a good singer, you better be exercising by singing all the time. True story? Do you guys now see that that is a true story? That it's the muscles, these tiny little minute muscles that are going to help create tension on those vocal cords. You ever hear of singers that sprain their singing muscles? They sprain their singing voice, like Adele, right? And they have to go in and get surgery to reattach or to help fix strained muscles. It's not a joke. It's real, especially if you're one of those professional singers. You put a lot of tension on these muscles. Just like any muscle, that means that it can be sprained or strained or broken. All right, cool. I probably spent more time on that than I needed to, but it's fun. It's fun. All right, last little bit. More. This is taking me longer than I wanted it to. I'm sorry. Last little bit is histology. Histology is the one that students tend to get the worst grades on semester after semester after semester. And that's because you don't get a specific histo picture. I don't give you a specific histo picture and tell you to label it, because that's not how histology works. Histology works by looking at dozens, if not hundreds, of different slides and different pictures and different depictions of things, and being able to identify it, because no histo slide is ever going to look the same. Doesn't matter whether you want to be a path. I know, I get the joke. You didn't even have to write ha ha ha. I get it. If you ask me who the smartest doctors on the face of the planet are, of all the doctors, there's a lot of smart doctors out there. But if you ask me who has to understand physiology and pathophysiology better than any other doctor, it's pathologist. Talk about a hard job where you take a biopsy from some person you know basically nothing about. You get a tiny little bit of history and you decide, in essence, what's going to happen to that patient. Everything lies on you. It's a crazy job. Respiratory system. You guys know about this slide box. You know, I shared it with you. Does anybody need me to share it with them again? I hope not. It's got everything, right? You want to go through and label nasal cavity, pharynx, larynx, trachea, the different bronchi, boom, they got that here for you, right? You can actually go through and look at what each one of those is. Sweet. What about nasal oral cavity? Well, you guys don't need to do histo on that. Epiglottis? No, we don't need to do histo on that. Larynx? Nope, I didn't give you that either. Trachea? Yep, definitely need to know trachea. I love the trachea, it's gorgeous. It looks like a rainbow. Esophagus, well, esophagus isn't where it's supposed to be up here. But, I mean, it's one of the most beautiful of the histological structures. Now, what I'm going to do is I'm going to keep zooming in. Zoom in. Zoom in. Zoom in. You guys see how I zoomed in on this dark purple zone? Yes, no, maybe so? I'm trying to go fast here. And as I zoomed in, what you started to see were more and more holes. I call these my googly eyes. Do you guys see the googly eyes? Googly eyes, googly eyes, googly eyes, googly eyes. Yeah, lacuna, lacuna matata, right? Ain't no worries. Lacuna. Perfect. Well done. So these are lacuna, lacunae plural. Lacuna means little lake in Latin, which is helpful. Okay? Cartilage cells, chondrocytes, they can't live in cartilage. Cartilage is freaking hard structure. There's no liquid. A cell couldn't possibly live in cartilage unless it surrounds itself with a microenvironment, a lake of fluid that it can live in. So these little open spaces are lacuna, or little lakes. Again, pretty easy to remember if you remember lacuna matata. Means no worries. It's its nutrients, gets rid of waste. We gotta. Oh, houseboats. God, I love those. Too bad they're so expensive. So now If I see lacunae, what do I know I have? I know I have cartilage. It's got to be cartilage. Is that clear? This is important. I'm not just doing this for shits and giggles. This is going to come back and haunt you if you're not paying attention. If you see lacuna, that's your landmark for cartilage. It's a really important landmark for cartilage. So now if I were to send you guys a picture for a lab practical and I asked you what layer is this, whether you understand that you're looking at the trachea or not, if you see all these little holes, especially if those little holes have little nuclei sitting in the middle of them, then you know this has to be cartilage. And therefore, what layer is this? Now we're starting to be able to use some logic. It's going to be a cartilaginous layer, okay? So when we go to our objectives, you're going to notice that you're going to have to be able to identify the different layers of the trachea. A mucosal layer, a submucosal layer, a cartilaginous layer, and something called the adventitia. You'll also have to find that trachealis muscle. So how do I know this is the trachea? Trying to give you guys the landmark that I used. This big purple ring, this C-shaped purple ring, is cartilage. Yes, no, maybe so? I just proved that to you. Mucus is produced on the inside of the tube. So where is the mucosal layer going to be found? It's this entire inside layer right here. So if I had to depict, goodness gracious, why is my mouse not working very well? If I had to depict where the mucosal layer is, it's going to be just this surface layer right here. And if I keep zooming in, zoom in, zoom in, zoom in. Actually, I might just be able to click respiratory epithelium. And that, yeah, there we go. If I keep zooming in, this is going to zoom in all the way onto that mucosal layer, the respiratory epithelium. These are simple, but not simple. They look like they're stratified cells. It looks like there's multiple layers of cells, but they're not. It's fake. So that's why this area right here is pseudostratified. You may or may not remember that word from A&P1, but we got these pseudo-stratified. Pseudo means false. So it looks like it's stratified epithelial tissue. It looks like there's lots of different layers, but there's not. It's false. If you guys want to go over that with that, want to go over epithelial tissues with me, you can meet up with me in office hours, okay? You want to learn about those again, I'm more than happy to do that, just not using time right now to do that. And then you can see that the surface of this looks fuzzy. Does it kind of look like it's fuzzy to you guys? Like it's got this light pink shade all the way out here? Those are cilia. So if I pointed to those, that would be cilia. Cilia of the mucosal layer. Last but not least, we have these kind of open spaces. I have one right here, one right here. They're not great. I'll try to find better pictures of this for the lab practical. I'm not finding it really fast here. But those open spaces, those open cells, let me just point this out online for you guys, are called goblet cells. Goblet cells are the unique cells of our epithelial tissue that produce, here's some intestines right here. These are large intestines. Open image in a new tab. And what you'll notice is that there's basically tons of these open spaces in our large intestines. These open spaces, each one of these open ovals is called a goblet cell, and they produce mucus. Goblet cells produce mucus. Can you guys write that down for me? Goblet cells produce mucus, which is important because the trachea produces tons of mucus, right? Well, let's get through that. The trachea does produce lots and lots of mucus. Now, the next layer, whoa, 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 whoa. What is it doing? It doesn't like me. What is it doing? I'm not pressing any buttons right now. My computer's on crack. All right, let's reboot this whole thing here. I just want to be done. Okay, here we go. So the way this works out is that we have a mucosal layer. I know, serious. Oh, I'm auto-tuning? Maybe that's why. But anyways, let's do this real quick. We got a mucosal layer, and then what's going to be underneath? What's going to be under the mucosal layer? What is all this dark purple stuff right here? Look at your objectives in case you're curious here, right? You should be looking through these. If I got a mucosal layer, what is underneath mucosal layer? Underneath. What is that? What layer is this going to be? Submucosal layer, good. And then inside of that, all of these swirls right here, these are glands. These are very, very important seromucus glands. They are going to help with the production of even more mucus. Anybody ever heard of the disorder cystic fibrosis? There's a lot going on down here in the trachea. Lots of excess mucus being produced, and it has to do with these glands and goblet cells and nasty. Next layer out is cartilaginous layer, and the very outside, of course, cartilaginous is C-shaped, so you're only going to see a portion of it. And then the very outside, if I point to the outside of this area out here, that is going to be helping to connect the entire trachea together using connective tissue, and that's called adventitia. Okay, so all of this outside loose connective tissue that helps hold it all together is called adventitia. Whew. Last but not least, I already showed this in the lecture, but if I have a muscle that holds the ends of the tracheal cartilage together, that is the trachealis muscle. It's just a smooth muscle. So it's this pink stuff, not the purple stuff. Purple stuff is glands. Those are seromucus glands. But this pink stuff is smooth muscle that helps hold those cartilaginous rings together. If you guys need to go, go. I'll stick around an extra couple of minutes. You guys can, whoever does need to go, just get out of here. And this is going to take me maybe 5-10 more minutes if you guys can hang out that long. But anybody that does need to leave, then you can come back and watch this part later. I feel bad. I wanted to let you guys out early today, but I got caught up later. So what I want you guys to see here, it's pretty cool. Same idea that we just went through. Do you guys see that there's a dark purple mass up here with holes in it for everybody that's left over? So dark purple with open space. What is this telling us? What is this? This is cartilage. And what did we learn about the tubes? No, not elastic. That tells us cartilage. So that, remember, if it's got the open space, then that means lacuna. Lacuna matata. You can start to see them even better as we zoom in. Got your googly eyes, really cool googly eyes, owl eyes. So that's all going to be lacuna. So that's cartilage. True story? Actually can't see the elastic fibers very well in any of these histo models. But just a quick point, remember we got our flow chart over here. Primary bronchi, secondary bronchi, tertiary bronchi, terminal bronchioles. What is happening between the bronchi and the bronchioles? What are we losing as we get down there? We lose cartilage. So if I see a tube, here's a tube right here. It's a big ass tube, this big open space, and it has cartilage around it. Is this a bronchiole? Good quick answer, Andy. Is it a bronchiole if there's cartilage that you can see around it? The answer is no. So what if I went down to this tube? Here's another tube. What is this? What is this? And what is this? Even what is this right here? It doesn't have to be super dark colored for it to still be. That's cartilage. Cartilage, 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 and it's surrounding this tube right here. So is this a bronchiole? Yes or no? No. Good. Fantastic. Well, now let's get down to this tube. This tube. Do you see any cartilage surrounding this tube? Anywhere? Is there cartilage anywhere around this tube? So, is this a bronchus? No, because bronchi have cartilage that surround them. So now that we have a tube with no cartilage, what do we know this must be? It's a bronchiole. Good. So this is a terminal bronchiole. There's one there, and there's one here. What makes these unique? No cartilage. Make sense? What about a bronchiole? Bronchioles aren't going to have much cartilage around them, if any. So this is a primary bronchiole. It's muscular, lots of muscles that surround it. You can see the muscles around it. Versus this down here, here's a tube with cartilage around it. So what is this going to be? Help me help you. Cartilage. Good. So this is a bronchi or a bronchus, singular. And that bronchus is now split, and it's finally small enough that it's starting to become a bronchiole. Does that make sense? Awesome. I'm really trying to help you guys because the lab practical, that's going to be your easiest way of identifying a bronchus versus a bronchiole. You just say cartilage, yes or no. So up here, what's this guy? Cartilage, yes or no? And if there's no cartilage, then you know it's a bronchiole. And that's good enough for your objectives. Now, what if there's cartilage? What if there's cartilage? What if there is cartilage? Easy peasy lemon squeezy. What do you know? Are there other ways to do this? Of course there are. Like if you zoom in all the way on a bronchus, the epithelial tissue is simple columnar. If you guys remember what that means. Versus if you do a bronchiole, if you zoom in on the bronchiole, we're going to start getting thinner. It's still simple columnar, but it's not quite as thick. And now when we get down to the terminal bronchioles, check it out. We're now starting to get almost a simple cuboidal. Does that make sense to you guys that we're going to have thick epithelial tissue that's going to get smaller and smaller and thinner? Love it. So now when we finally get out to the alveoli, okay, and the respiratory bronchioles, now we're looking at tubes that are almost impossible to see as tubes. But there is some simple cuboidal, and it turned into simple squamous. Now we don't have this thicker epithelial tissue. Now it's just turning extremely thin. Yes, no? We're going to be going from something that's cuboidal, and we're going to turn into something that's not. But you guys don't need to be able to identify respiratory bronchioles on the lab practical. You don't need to go into that much detail. I'm just trying to get you guys to see that it gets thinner and thinner and thinner. And finally, the respiratory bronchioles are going to be emptying into ducts. So how do I tell the difference between a duct and an alveolus? This isn't actually a great one, but it works. Do you guys see how this is kind of open all the way through? Open, open, open. It's kind of big open space, bigger open space here, but it's not an individual circle. It's not an individual circle. Yes, no, maybe so. There's an individual circle here, but this is not an individual circle. So as we're looking out towards especially the borders out here, you're going to notice there's a lot more individual circles, versus some open spaces. Open. This is a pretty good depiction of one right here. Open space where it's almost like a maze. Those mazes are going to be the alveolar ducts, okay? Versus the individual circles, which are alveoli. Ish, kind of makes sense? Cool. I think that's kind of your objectives. Bronchi, bronchioles, that's the one that students have the most difficult time with. Alveoli, alveolar duct. I just realized, oh shit, I didn't know this. You need to have alveolar duct on this list. I don't know why it's not here. Can you make sure that your lab partners know? Cannot just have alveoli, we also need to be able to identify the alveolar ducts. I'll try to send out, somebody might be able to remind me of that. Pulmonary arteries versus pulmonary veins. I'll let you guys do a little bit of research into that, but just try to remember what their functions are. It's a little bit tricky because of the pressure gradients, but yeah, I'll deal with that later. It's 32 after. I feel so bad that I've kept you guys so long. Welcome to summer. Probably not a lot of immediate questions. Are there any immediate questions while I'm here? By the way, so if you are gonna, let me just do, okay, thanks, Andy, thank you. I mean, I could stay a long time, but it is what it is. I don't mind helping. But let me get you guys a hint on finding pulmonary arteries versus pulmonary veins. Anybody recognize what I'm zooming in on? What are these little inner tubes? Well, my daughter just went down for her nap. My wife has today off from work, and they just went, I don't know where. She handed me the monitor and said they were going out. So I'm not sure where they're going. They just got back from the park too. But what am I looking at here? What are all these little inner tubes? This should be familiar, oddly familiar to you guys. You should be having some deja vu. What am I looking at? And I got a few cells in here that have these purple nuclei. Yeah, thank you, Ashley. These are erythrocytes, right? And these, mostly in this picture, these are all neutrophils. And I think that's even a neutrophil there, maybe a lymphocyte. Oh yeah, a lymphocyte right there. There's a monocyte down there. But yeah, this is blood. So now if I zoom out, if I got blood, then I'm probably inside of a gigantic blood vessel. True story? You can see that this blood vessel here looks different than this blood vessel here. Different than this blood vessel here. Different than this blood vessel here. This blood vessel here looks a little bit like this blood vessel here. It's got this kind of purple border, big, thick purple border, versus these blood vessels out here don't have that big, thick purple border. So one's going to be an artery and one's going to be a vein. I'm going to let you guys figure out the rest. Okie dokies, artichokes. All right, you guys rock. Thanks for sticking it out. No. Thank you. Don't thank me. You guys, it's hard to keep your attention for this long. I understand, and I'm sorry, but I can't. Just as an end note, I can't be that professor that says, "Here's your objectives list, go." Like, I just can't bring myself to do it. So I apologize that sometimes I spiel too long. That's because I actually want you guys, I feel like it's because I actually want you guys to learn. And then if you just give kids a objectives list, the reality is they're not always going to go and find what they need to find to be successful. So, all right, guys. Reach out if you have any questions, if you need any help, if you want to go through different parts of this, just let me know. All right? You're welcome. We've gone through every single objective together today. When is this due, by the way? When's the extra credit due? Speaking of which, you guys rock. I'll open it up right now. Respiratory anatomy. Those will be due Wednesday at midnight. All right, get on out of here, y'all. I'll start grading your exams soon, and should have them done by the end of the week, just like I had last time.